University, Hyderabad. This conference serves as a platform for researchers, academicians, industry professionals, and students to engage in meaningful discussions, exchange groundbreaking ideas, and foster collaborations that will shape the future of healthcare.
Over the course of 3 days, we will witness enlightening keynote addresses, expert lectures, and innovative research presentations that contribute to advancing healthcare solutions globally.
To set the perfect tone for the conference, we begin with a mesmerizing welcome dance by Swapnika, our B.Pharm students.
I would like to invite Swapnika, our B.Pharm student.
Shiva Shambho Swayambho Bho Shambho 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 Shiva Shambho Swayambho
Thank you, Swapnika. That was truly a mesmerizing performance.
To begin this grand event on an auspicious note, we invite Dr. Shrikant Gatadi to present an invocation song. His soulful performance will invoke positivity, harmony, and setting the perfect tone for inspiring sessions ahead.
Good morning, Namaskar. So let us all enjoy this beautiful Bhakta Ramadasu Sankirtana. Yes. పలుకి బంగారమాయేనా పలుకి బంగారమాయేనా పలుకి బంగారమాయేనా కోదండపాని పలుకే బంగారమాయేనా పలుకే బంగారమాయే పిలచిన పలుకవేమి పలుకే బంగారమాయే పిలచిన పలుకవేమి కలలోని నామస్మరణ మరువా చక్కని స్వామి పలుకే బంగారమాయేనా కోదండపాని పలుకే బంగారమాయేనా ఇరవుగ ఇసుకలోన పొరలిన ఉడత భక్తికి ఇరవుగ ఇసుకలోన పొరలిన ఉడత భక్తికి కరుణించి బ్రోచితివని నెరనమ్మితిని తండ్రి కరుణించి బ్రోచితివని నెరనమ్మితిని తండ్రి పలుకి బంగారమాయేనా కోదండపాని పలుకే బంగారమాయేనా ఎంత వేడినా గాని సుంతైన దయారాదు ఎంత వేడినా గాని సుంతైన దయారాదు పంతము సేయు నేనంతటి వాడను ఓ తండ్రి పంతము సేయు నేనంతటి వాడను ఓ తండ్రి పలుకి బంగారమాయేనా కోదండపాని పలుకే బంగారమాయేనా ಶರಣಾಗತತ್ರಾಣ ಶರಣಾಗತತ್ರಾಣ ಬಿರುದಾಂಕಿ ತುಡವುಗಾದ ಕರುಣಿಂಚು ಭದ್ರಾಚಲ ಪರರಾಮದಾಸ ಪೋಷ ಕರುಣಿಂಚು ಭದ್ರಾಚಲ ವರರಾಮದಾಸ ಪೋಷ ಪಲುಕೆ ಬಂಗಾರಮಾಯನ కోదండపాని పలుకే బంగారమాయన కోదండపాని పలుకే బంగారమాయన పలుకే బంగారమాయే పిలచిన పలుకవేమి పలుకే బంగారమాయే పిలచిన పలుకవేమి కలలోని నామస్మరణ మరువా చక్కని స్వామి పలుకే బంగారమాయన కోదండపాని పలుకే బంగారమాయన కోదండపాని పలుకే బంగారమాయన. థ్యాంక్ యూ ఆల్.
Thank you, Dr. Shrikant Gattardi, for that divine and uplifting performance. Your melodious voice has truly added a spiritual essence to our gathering.
Now, it is time to extend a grand welcome to our dignitaries. We request the following distinguished guests to kindly grace the dais. Dr. Pratik Pathak and Dr. Ashish Ranjan Dwivedi, convener of Healthcare Next 2025. Sir, please come on the stage.
Now, I would like to invite Professor G. Shivakumar, Principal, Geetham School of Pharmacy, Hyderabad. Sir, please grace the dais.
Now, I would like to invite Professor Jagataran Das, Dean, Geetham School of Pharmacy. Sir, please.
Now, I would like to invite Professor D.S. Rao, Pro Vice Chancellor of Geetham University. Sir, please grace the dais.
Dr. Akela Venkateshwarlu, Founding Director, Dr. Reddy's Institute of Life Sciences. Sir, please praise the dais.
I would like to request Dean Sir and Pro VC Sir, please present a token of appreciation to the Venkateshwarlu Sir. Now, I would like to invite Dr. A. Ramkishan, Deputy Drug Controller, CDSCO, Hyderabad. Sir, please grace the dais.
We are truly honored to have such eminent personalities among us today. Thank you all for gracing the dais and lending your presence to this prestigious event.
To mark the beginning of this event with positivity and wisdom, we now proceed with the traditional lamp lighting ceremony. We kindly request our esteemed dignitaries to join us in the lighting of lamp, knowledge and enlightenment. Thank you to our respected dignitaries for this auspicious begin.
To formally begin with the event, we now invite Dr. Pratik Pathak, convener of Healthcare Next, for their welcome address.
Honorable dignitaries, esteemed speaker, distinguished guests, respected university authorities, dear colleague, and all the students. It is with immense pleasure and honor that I welcome you all in the International Conference on Integrative Trends in Pharmaceutical and Health Sciences, term Healthcare Next 2025. As the convener of this event, I extend my heartfelt gratitude to each one of you joining us today. At the outset, I would like to express my sincere appreciation to our university leadership, including our Honorable President, Honorable Chancellor, Honorable Vice Chancellor, Honorable Pro-Vice Chancellor of Hyderabad campus for their support and vision for fostering academic excellence and research collaboration. In their guidance has been instrumental in making this conference a reality. A special and warm welcome to our chief guest, speakers, distinguished guests, and other researchers whose presence today at this event a great value. Your expertise and contribution to this field of healthcare and pharmaceutical science are truly commendable, and we are privileged to you have with us. I am pleased to inform you that this conference will feature 60 plus oral presentation, 100 plus poster presentation, 15 expert lectures, and cultural events that will provide an enriching experience for all the participants. We are truly honored to have a diverse array of speakers from across the globe, including USA, Croatia, Malaysia, Sri Lanka, and India. The wealth of the knowledge will surely inspire and provoke new ideas as we delve into an evolving landscape of healthcare and pharmaceutical sector. It is also a privilege to host 450 participants from 3 different countries and 6 Indian states, which reflect a broad interest and engagement in this field. This international collaboration enriches the discussion and fosters a global perspective to the challenges and opportunity in healthcare sector. To my fellow colleagues and research scholars, I deeply acknowledge your dedication and relentless pursuit of scientific excellence, your effort in advancing healthcare through innovation, research, and collaboration are shaping the future of medicine and patient care. And to our young scholar and students, you are the driving force of the society which can create a next generation for the healthcare profession. This conference presents a golden opportunity to learn, engage, and inspired by some of the most brilliant minds in this field. I encourage you to actively participate, absorb the knowledge, and forge the meaningful connection that will shape the academic and professional journeys. The theme of this year conference, Healthcare Next, is a testament to our collective commitment and integrative traditional wisdom with modern innovation. As a naive through an era of rapid scientific advancement, it has become imperative to explore the transformative approach that will make healthcare more accessible, sustainable, and patient-centric. Over the few next decades or few next days, we will witness through provoking discussion, groundbreaking research presentation, and insightful deliberation that will drive the future of healthcare and pharmaceutical science. Let us use this platform to exchange ideas, foster collaboration, and work toward innovation or innovative solution for the benefit of the humanity. With that, I extend my warmest welcome to each one of you and wish you a profound, fully engaging, and inspiring conference. Thank you very much.
Thank you, Dr. Pratik Pathak, for your warm and insightful welcome address. Your words have truly set the stage for what promises to be an enriching and inspiring event.
Now we are privileged to introduce Dr. Akela Venkateshwarlu, Founding Director, Dr. Reddy's Institute of Life Sciences.
Dr. Akela Venkateshwarlu is a distinguished organic chemist and the founding director of Dr. Reddy's Institute of Life Sciences. With over 30 years of experience in drug discovery and pharmaceutical research, he has held senior executive positions in renowned companies, including Dr. Reddy's Laboratories, Novartis, and Aventis. A Harvard-trained chemist, Dr. Venkateshwarlu has made significant contributions to both academic and industrial research. With numerous patents and publications to his name, he had played a pivotal role in shaping the growth of several institutions, including Dr. Reddy's Research Foundation and Origin Discovery Technologies.
Dr. Akela Venkateshwarlu has an impressive academic background with a bachelor's degree in chemistry from WGB College, Andhra Pradesh, a master's degree in organic chemistry from Andhra University, and a PhD in organic chemistry from Wayne State University and University of Pennsylvania. His postdoctoral research at Harvard University under Nobel laureate Professor E.J. Corey further strengthened his expertise in organic synthesis. Throughout his career, Dr. Venkateshwarlu has been at the forefront of drug discovery and R&D management, contributing to the development of innovative therapies in the pharmaceutical industry. He has also played an integral role in advising emerging companies, serving as a member of scientific advisory team of Curious Therapeutics. His leadership and contribution have earned him recognition in both academic and industrial circles, making him a respected figure in the global pharmaceutical and life sciences community.
So now it's my pleasure to invite Dr. Venkateshwarlu to come forward, and we request Pro VC Sir, Dean Sir to present a token of appreciation to Dr. Akela, and I request the other dignitaries to present on the dais to join the felicitations. Now, we are privileged to invite Dr. A. Ramkishan, and I request Pro Vice Chancellor Sir and Dean Sir to present a token of appreciation to Dr. A. Ramkishan. Dr. A. Ramkishan is the Deputy Drug Controller General of India at the Central Drug Standard Control Organization, a regulatory body under the Ministry of Health and Family Welfare, Government of India. He has a distinguished career in pharmaceutical regulation and public health, with over 2 decades of experience in drug safety, quality assurance, and policy development.
Dr. Ramkishan holds a Bachelor of Pharmacy, Master of Pharmacy, and Doctorate in Pharmacy from Warangal. He joined CDSO in 1998 as a drug inspector in Mumbai and has since worked in various capacities, including Assistant Drug Controller in Ahmedabad. His leadership and expertise led to his appointment as a Deputy Drug Controller at CDSO, New Delhi headquarters from 2013 to 17. Currently, he serves at the Hyderabad Zonal Office as CDSO, where the continues to play a key role in overseeing drug regulatory activities across the country. Ramkishan's vast experience and dedication to improving public health have made him a respected leader in the field of pharmaceutical regulation, both in India and internationally.
Sir, we are honored to have you with us today.
Moving forward, we are honored to hear from Professor Jagataran Das, Dean Geetham School of Pharmacy, Hyderabad. With his vision and leadership, Geetham continues to be a hub for innovation and excellence in pharmaceutical sciences. Sir, we invite to share your thought with us.
Hello. Is it audible? Honorable Chief Guests, Dr. A. Venkat Sarlu, Founding Director of Dr. Reddy's Institute of Life Sciences, Dr. A. Ramkishan, Deputy Drug Controller, CDS CEO, Professor D.S. Rao, Pro Vice Chancellor of Hyderabad Campus, the Principal and the 2 coordinators, conveners of this conference from Geetham School of Pharmacy, distinguished scientists, esteemed colleagues, generous sponsors, and my dear students. A very good morning to all of you. It's with great honor and excitement that I stand before you today at the inauguration of the International Conference on Integrative Trends in Pharmaceutical and Healthcare Sciences, Healthcare Next 2025. This momentous occasion marks not only a significant milestone for the pharmaceutical and healthcare sectors, but also an exciting opportunity for all of us to come together, collaborate, and take the innovations forward to shape the future of healthcare. Last year, we successfully hosted the International Conference on Healthcare Next 2024 at our Visakhapatnam campus with enthusiastic participation of the students and faculty members from Hyderabad campus. Following this success, we decided to make Healthcare Next an annual event, rotating the conference between the 2 campuses, ensuring that our students and faculty have the opportunity to connect and collaborate beyond scientific discussions. This year's Healthcare Next 2025 marks the continuation of this series of international conferences, and I am pleased to share that over 80 students and faculty members from the Visakhapatnam campus are actively participating in this conference. This conference offers a unique opportunity to engage with some of the brightest minds in the field, share cutting-edge research, and explore innovative solutions to the challenges facing the healthcare sector today. The integration of cutting-edge technology, novel drug therapies, and patient-centered care models is reshaping the very foundation of healthcare industry. From personalized medicine to artificial intelligence in drug discovery, we are on the cusp of breakthroughs that will redefine the way we approach treatment, prevention, and healthcare management. In this dynamic environment, collaboration is the key. The exchange of knowledge, experience, and ideas between experts and the next generation of professionals, particularly our students, is essential in ensuring that we continue to push the boundaries to achieve our goal. I encourage each one of you to actively participate in the discussions and networking sessions throughout the event. Let us share knowledge, foster meaningful collaborations, and inspire one another to create a more equitable and healthier world for all. In closing, I warmly welcome all delegates, speakers, and participants to this enriching conference and extend my gratitude to our dedicated faculty, staff, and students whose efforts have been pivotal in organizing this event. Let's embrace this opportunity to significantly contribute to healthcare advancements. I wish all the success for this conference. Thank you very much.
Thank you, Professor Jagataran Das, for your inspiring words. Your leadership and dedication to academic excellence continue to drive innovation and progress at Geetham School of Pharmacy. Thank you so much, sir.
We now move on to a significant moment of the event, the abstract book launch. This book is a compilation of insightful research and innovative ideas that will shape the future of healthcare and pharmaceutical sciences. To formally unveil the abstract book, we invite all the esteemed dignitaries on stage to join this special moment.
We request Dr. Pratik Pathak, Dr. Ashish Ranjan Dwivedi, Professor Jagataran Das, the Pro VC Sir, Dr. Akela Venkateshwarlu, Dr. A. Ramakishan, and Professor G. Shivakumar to kindly proceed with the book launch.
Thank you, esteemed dignitaries, for unveiling the abstract book. This moment symbolizes our commitment for research, excellence, and knowledge, dissemination in the field of healthcare and pharmacy. Now, we are privileged to invite Dr. Akela Venkateshwarlu, Founding Director, Dr. Reddy's Institute of Life Sciences, to deliver the inaugural address.
It's my pleasure to invite Dr. Venkateshwarlu to come forward and share his insights with us. Let's give him a big round of applause.
Hello. Am I audible? Hmm?
Hello, hello, hello.
Hello. Hello. కొంచెం. Hello. 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 Okay. Namaskar. Once again, namaskar to the audience. Distinguished Vice Chancellor, Professor G. Sambasiv Rao, my fellow scientist Jagataran Das, distinguished participants in this conference, students, ladies and gentlemen. When Jagataran asked me, rather came to invite me to grace this occasion and address the audience, I have readily accepted, not knowing that I will undergo a surgery. But having accepted and keeping the scientific spirit, I managed to come here. Instead of stand before you, I am seated and then give my message. I did not know much about Geetam, although I have known such an institute existed in Visakhapatnam. But after this invitation, I read much about Geetam. I must profusely congratulate the founders, the faculty, and the management of this great institute. Pharmaceutical education, drug discovery are close to my heart. I have been in this field for the past 45 years. The main theme of this conference, integrative trends in pharma and healthcare sciences, Healthcare Next 2025. This theme of this conference and the subject intent of this event is certainly beyond the scope of my comprehension. Although I have practiced medicinal chemistry, research, drug discovery in particular, and working in an interdisciplinary environment in GSK, Siva Gaige, which is now Novartis, and Dr. Reddy's laboratories here in Hyderabad. If you go back 50, 60 years in this drug discovery game, we mostly chemistry, pharmacology, toxicology. And of course, regulatory framework which was in vogue at that time. Look at the discovery of aspirin. One scientist in Bayer has stumbled upon the synthesis of acetyl salicylic acid, which they named aspirin. Even then, it took more than 2 years to bring acetyl salicylic acid in the form of aspirin into human medicine. But many of you know, aspirin also is a natural product. It's derived from willow tree, perhaps today world's largest produced pharmaceutical ingredient. Those many discoveries originating from natural products. There was no interdisciplinary effort, chemistry, pharmacology, toxicology. Outstanding discoveries were made those days. But the first rational drug discovery, to my mind, appears to be cimetidine by Smith, Klein and French. It is sold under Tagamet. It was the first billion-dollar pharmaceutical ever known to mankind. It was followed by a few years later, almost the same team of Sir James Black, discovered ranitidine in Glaxo, in UK. When cimetidine was discovered, Smith, Klein and French wanted to bid for Glaxo. Both are in UK, Smith, Klein and French, UK. But when Glaxo discovered ranitidine, they wanted to acquire Smith, Klein and French. Cimetidine was $1 billion molecule. Ranitidine became $3 billion molecule. Cimetidine was outdated because of some carcinogenic property surfaced after 3 or 4 years of introduction. Ranitidine appeared to be still a high flyer. Even ranitidine came into problem because of NMDA nitro issue has put it in behind the desk. Then came a family of new drugs in the form of omeprazole. Those were H2 receptor antagonists, cimetidine, ranitidine, nijatidine, so on and so forth. But these are proton pump inhibitors. Omeprazole came as a boon to the people, and it has replaced the therapeutic profile of ranitidine, H2 receptor antagonist. I don't want to go into the actual discovery. I have known scientists who discovered these molecules. I was working in Smithkline in French, and I have known team which went out into Glaxo, and I have interacted with the pharmacologist, and I had the fortune of meeting Sir James Black, who was very scientist, with very difficult to manage. But many antibiotics, microbial metabolites, became star discoveries. Waxman discovered streptomycin. Before that, penicillin and all that. After microbiology came, ever since the discovery of nucleic acids, structure of nucleic acids, the advent of molecular biology, subsequently molecular pharmacology, then genetic engineering, so on and so forth. These are all rapid advances that have come into the scientific realm of drug discovery and development. But present day, this integrative, earlier they said interdisciplinary, then became multidisciplinary. Now you have integrative trends in pharma and healthcare sciences. This is mind-boggling. So my grandson asked me once, how did you manage without 4G, 5G? I was stunned. And I told him we lived in times where we worked with only 3G. Mataji, Pitaji, Guruji. That much a dramatic difference you see in today's drug discovery and yesteryears. I participated in somewhere in between. Drug discovery at Dr. Reddy Drills, Dr. Reddy's Institute of Life Sciences, young workforce like Parimal, Jagataran, Nanduri, and many young scientists who have returned from abroad looking for industrial position in research. This was the first example of an interdisciplinary team that was put together in Dr. Reddy's Institute of Life Sciences. At peak times, we had something like 45 to 50 PhD scientists drawn from medicinal chemistry, in bracket organic chemistry mainly, natural product scientists, pharmacologists, toxicologists, and even clinical scientists. But those days it was adventurous on our part. The man behind all this is a force by name Dr. K. Anji Reddy. Drug discovery was so much obsessed with this field. He used to tell us, if we don't discover and develop new molecules now, we will be driven back to tribal times. This was the word. That sentence itself inspired me to leave multinational labs and come to Hyderabad and join his. Perhaps by the same spirit, I could attract young, bright scientists, 35 to 45 PhDs. There are MTechs, chemical engineers, every field that is required to put together elements of organic chemistry and molecules to bring out 3 drug candidates that have reached clinical trials. It's mind-boggling in the first period of 7 years, bringing 3 molecules, not only to clinical table, but also to licensing table. We have earned $13 million by upfront payments and all that. Unfortunately, drug discovery and development is very risky. Those days statistics were, it would have cost something like $1 billion plus. And it would take 10 to 12 years and at least 200 to 300 molecules. Many of them fail. 10 molecules reach clinical table. Only 2 or 3 survive the rest of it. And even those molecules, rapid obsolescence. That's the characteristic of drug discovery research. Under those circumstances, we have learned a lot of intricacies of drug discovery and development. I have learned and mastered recruitment of right scientists. This is very important. Checks and balances, a mix of interdisciplinary mindset, and people who could think and work like team members. At Dr. Reddy's, I used to tell people, said, how we borrowed in first 5 or 6 years so many molecules. The reason is each person is doing not one plus a delta. If 50 scientists, I have 50 times 50 deltas. This is how synergistic workforce made it possible to bring out those molecules in a short period. Today, these integrative trends in pharmaceutical and healthcare sciences, there is another dimension, artificial intelligence. It's so fantastic. Artificial intelligence is going to play a great role in drug discovery and development. Today's drug discovery and development is completely different. I happened to visit a modern lab now. I was pleasantly surprised. None of those machines I had seen during the time I practiced. This science at Harvard, which is the top of the academic world. It's fantastic. With only 15, 20 scientists, you can make the discovery. Earlier, we used to take 30, 40 team members. I'm too old now to practice those things, but I'm very happy to read and comprehend with notes given by Nanduri, Parimal, and other modern scientists. Coming to my slide talk, this may be few months back, I came across a book called Mission Possible. In bracket, Universal Healthcare. Incidentally, this book was written by the Vice President of Clinical, Swaminathan Subramaniam, I had recruited at Dr. Reddy's. He lives in Bangalore. He had an MTech, MBA as co-author. MTech in Chemical Engineering, IIT Madras, IIM Bangalore, MBA. They both have written this book called Mission Possible, very thought-provoking and catchy. I went through some pages of it. There's not much science in the book. The framework that helps bring healthcare to the human patients. So I thought I am not able to talk so much about these integrative trends in pharma and healthcare sciences. However, the fruits of this research should address this problem, universal healthcare. It could be interpreted as two ways. Universal healthcare or universal let us say universal healthcare. What does it involve? It only uses the science that comes out of this interdisciplinary or integrative trends in pharma and related sciences. The mechanism, administrative support, and governmental involvement can only bring these medicines to the mankind. Now today you see, the more interdisciplinary it is, more multidisciplinary it is, most expensive drugs, more the expensive drugs. Take for example, rare diseases which are intractable. There are 7,000 rare diseases known and hardly medicines for 50 or 60. Something like SMA you must have heard, spinal, muscular atrophy. Some people call it dystrophy also. Novartis came out with a gene therapy. It's called Zolgensma. You know what it will cost? $1 million. Can we afford? Nobody, unless there is an insurance, governmental support. SMA, similarly DMA, Duchenne's muscular dystrophy. These are all very intractable diseases. And people who are affected by this, it's a genetic at birth this come. What will happen? They live for 7 or 8 or 10 years when they die because there is no effective medicine. Gene replacement therapy took so long for Novartis to bring it, and then it was unreachable. You have a beautiful fruit on the tree, but by the time you pluck it will. How do we manage this kind of situation in future? There are 7,000 diseases. Maybe about 60, 70 of them are very dangerous. A baby born with such a disease is dependent on another person. So one person is already invalid. The other person's productivity for the society is tied up with this patient. It's horrible. So many other diseases, thalassemia is there, phenylketonuria. These are all simple. We are able to manage, prolong the life of the kid despite handicap. So these mechanisms of, drugs controller department is so important for regulating marketing and use of any pharma. When it comes to healthcare that could not be addressed, imagine what kind of framework will require. If you see the first slide here, what is universal healthcare? All of you can understand. This is UHC ensures access to health services required for humanity, regardless of economic and social status. A rich man can go and buy, but even rich man will find it difficult if it is a $1 million drug. So, there are several other features described here to define what UHC means. Most important thing is, in summary, universal health care ensures accessibility, affordability, and quality of patient-oriented health service. What it means, the economy does not matter, society does not matter. At the given time, medicine should be made available. It looks like Ramraj. I don't know when we see this. But if you read this book, we are perhaps within a decade or two to put at least 50% of the requirements of UHC. Out of 195 countries, except 43, all others have some kind of UHC, satisfactory. Even the wealthiest, mightiest country like United States does not have UHC. It is a capitalist country. So they have not, nothing is free in America. You have to pay and get it. Even small country like Norway, Denmark, Sweden, there I understand from birth to death, pharmaceuticals, medical requirements are taken care. Education and these are taken care. But they pay 40-50% income tax. So in a country like India, imagine, we are mind-boggling. We have 1.3 billion people. Very few, even the very rich people could afford to reach medicines like this. The rest of my slides say It says economic and social benefits of UHC. Second slide. Second slide. No. Economic and social benefits of UHC. Can we change? No. Second one. Yeah, this is the most populated. So here you see what's happening. Countries with UHC have lower mortality. What are the benefits of UHC? Countries which practice UHC have lower mortality rates and higher life expectancy. Preventive care reduces the burden of chronic diseases. In some countries, the so-called genetic disorders, they want to eliminate wrong genes from the genetic pool. How do they do it? Once they see a baby is born with a genetic defect, they invite both parents and counsel them not have any more kids. Or if they have any kids, some are carriers, some kids will get the disease, the others will get carriers. Whatever carriers can be there, but one who affects is removed from the genetic pool. This is how they have achieved in some countries very low percentage of genetic errors at birth. In our country, even though you know that a kid is born with genetic disease, they will go to temples, they will go pujas, they will do so many things. It's not going to alter. We are suffering. If all of us are here to listen to this lecture and comprehend and think further, we are very fortunate. Genetic errors is such a my wife is a geneticist. She is a children's specialist specializing in genetics. Every evening she comes disheartened. You know that there is a genetic disorder, but you don't have a medicine to produce. Simple things are there. Recently, one pharmaceutical company came out with 2 such medications, and they are giving on compassionate grounds subsidized prices. So preventive care is another, that's what people have in other countries are doing. They will do genetic counseling of both wife and husband and advise them appropriately to reduce the genetic burden in the society. Of course, early intervention leads to, yeah? No? 5 minutes. Early intervention leads to long-term savings. Other benefits are enhanced productivity due to better health. This is one of the benefits of UHC. Improved social stability and equity. I have only 2 more slides. Use of vaccines have brought great relief for mankind. You must have seen COVID times. Otherwise, half of the mankind would have perished. Women's health and therefore access to parental and postnatal care, child health, immunization that we have against polio and other serious disorders, nutrition and early intervention protocols, improve survival rates. There are challenges. Of course, financial constraint is one thing, sustainability is another, political issues such as government subsidies and all that, you are not, poor versus rich, limitations in infrastructure and trained workforce. Artificial intelligence, digital healthcare will bring some more efficiency in this field. Telemedicine, of course, to bridge the rural and urban gaps. Personalized medicine in preventive care. In conclusion, universal health care is not just a another slogan that you could use in healthcare maintenance, but it is a moral, economic, and scientific imperative. Investing in healthcare leads to long-term societal benefits. Need for continued efforts to promote UHC via research, innovation, and policy advocacy. When I first read this title, you must have heard universal healthcare before. I had not heard before. First time I read Mission Possible. This is eye-catching. And this is fantastic. I don't know how many of us will live to see the beneficial effects of UHC. But maybe these integrative trends, the theme of this conference, should contribute towards bringing medicines that are affordable to help UHC implement in a developing nation like and populous nation like India. Thank you very much for your patience. Hope some of us will live to see this, some elements of UHC implemented, and all scientists should feel happy that they will contribute to this mission in India. Thank you, sir.
Thank you, Dr. Akela Venkateshwarlu, for your inspiring and thought-provoking address. Your insights have truly set the stage for meaningful discussions ahead.
Now,
we thank you once again for being here with us, for sharing your wisdom and for inspiring us all.
now we take this opportunity to invite Dr. A. Ramkishan. Now we take this opportunity to invite Dr. A. Ramkishan, Deputy Drug Controller, CDSCO, Hyderabad, to deliver his inaugural address and to provide us the insightful lecture. Sir, kindly grace the dais.
Hello. Is it audible? Yeah. No, it's not required. Chief patron of this international conference on integrated pharmaceutical and health sciences, Sri M. Sri Bharat, the president and management personality from Geetham University, an honorable member of parliament, Visakhapatnam, Andhra Pradesh, and co-patron, our pro vice chancellor, Sri D. Sambashivarav Garu, and chief guest, Dr. A. Venkateshwara Garu, the founder director of Dr. Reddy Laboratories Limited, Hyderabad, then Andhra Pradesh, now Telangana, and the principal, Sri Shivkumar ji, and our respected Das ji from Visakhapatnam, and all faculty members, esteemed speakers, research scholars, part-time and regular, budding pharmacists, special invitees, and press personnel from electronic and print media. I am very glad to attend this important international conference. I am thankful to the management, particularly Sri Shivkumar ji, and the organizing committee, Dr. Pathak ji and Pratik ji. Please give a big hand to them, the way they have taken care since last 1 month. How really they have conglomerated with respect to this important, converting their thoughts. The objective of this conference is really, sir, you have narrated since 1909 to 2025, Dr. Venkateshwara ji, the way the regulatory path with respect to innovation, maybe cimetidine, maybe ranitidine, maybe captopril, maybe Alzheimer's disease in those days. Now things are changing, innovations are going in different directions. That is the reason why the theme of this Congress really kept in such a way that how we can integrate with respect to the transnational research, modern healthcare, and traditional healthcare. So this is how we can fill up the gaps, and really this will serve as one of the important platform for all esteemed research scholars to excel in their future with regard to drug development. So patient needs good product, and you have rightly pointed out 7,129 diseases as per the global data. I am also one of the expert committee member looking after all WHO guidelines, product development on behalf of World Health Organization as a regulator, as a part of regulatory strengthening. World Health Assembly resolution passed in the year 2014, 67.20 resolution, where the capacity building and then continuous training, and this is one of the important conference really putting efforts and to address the global challenges, how we can take care with respect to innovation. And having theoretical knowledge doesn't make any sense. How you can translate into the practical application to serve the vulnerable population as far as access, affordable, high quality medical products are concerned. This is number one. And we are from regulatory side, Government of India changing and taking lot of efforts with respect to simplification of the regulatory paths. When we talk about the clinical trials, then health research, and how we can promote the innovator to get into a real world solution with regard to availability of the novel therapies. We are having so many important diseases, so how we can target. So 85 diseases are very dangerous out of 7,129 diseases as per the global data. So things are changing, and research scientists are putting efforts globally, but still we need to have the holistic approach with respect to the filling the gap between the academicians, industry and the regulator. This framework, please sir, please madam, hello. Please spend 5 minutes, my dear friend. So how you can integrate with respect to this important interface is one of the important challenge. This is number 2. And Government of India, Ministry of Health and Family Welfare published one of the best, I can say, well-defined comprehensive text with respect to promoting clinical trials in India. We are not inferior to any country. India is taking lead with respect to promotion of the clinical research in human beings, how we can take care and what are the global challenges with respect to US FDA, PMDA Japan, ICS countries, EMA, European countries and UK MHRA, how we can integrate. So we are not having any inferiority. That's what I want to be very clear with respect to all research scholars friends. You can take lead. India is committed to promote the research with regard to pharmaceutical products. This is number 3. Number 4, we are having good Gazette notification issued on 19th March 2019 with respect to new drugs and clinical trials. This is one of the best texts based on the Honorable Supreme Court of India, and the Supreme Court bench gave the clear path with regard to informed consent process, investigator responsibilities, ethics committee responsibilities, sponsor responsibilities, then contract research organizations, CROs, how they have to take care with regard to meeting the good clinical practices. Olden days gone, we are number 1. India is going in different direction. This credit goes to our Honorable Prime Minister of India. Now things are changing and globe is looking for India. And in the year 1905, things were different. Dr. Anjali Sahab, how sir has developed and promoted the generic product. You see, highest number of drug master file in our country after United States of America. So things are changing. Everyone needs to have the high quality medical product without compromising regulatory framework. This is number 4. Number 5, our Nanduri Srinivas sir is here and he is also eminent personality. How sir came from USA and serving in the National Institute of Pharmaceutical Education Research, Naipur, Hyderabad. So we need good scientists. Only thing is we have to avoid ego so that we can bridge the gap. If we can avoid the ego, we can do wonderful things. India has excellent talent pool, excellent academicians like Geetam, excellent English-speaking students and all important people, excellent judiciary, excellent our important scientific people. Only thing is we need to have the gaps and how we can fill up. We can do wonderful things. 175 important molecules are patented. But what is the use? Unless you go for the this thing commercialization. Having some claims from the Government of India, Patent Office granting the patent and keeping in the cupboard with all certificates. What is the use? So just my humble request to all esteemed academicians here, please try to focus on commercialization. Having claims, granting US patent and Indian patent doesn't make any sense. My appeal, regulators also changing mindset of, we are acting like enablers, so there is no any ambiguity. This clear message, I want to give it to everyone. And Venkateswar Saab, now mindset of regulators also changing. We are having 95% of the e-governance is in place. Olden days gone. Now we are not having any ambiguity. So things are changing. We are all with you. We are with Indian people. And ultimately, we have to develop novel, important therapeutic products to serve the vulnerable population for ensuring quality, purity, identity, safety, and efficacy of all important medical products moving in international commerce to achieve patient safety for the promotion of the global health. So I will not take much time. Next time I will come with the presentation, but my best wishes. I am very happy to grace this occasion in front of all important eminent scientists, and thankful to Shiv Kumar ji and the faculty members and Pro-Chancellor D. Samshiraji and all the research scholars and budding pharmacists. Thank you very much. Value as a value, only if its value is valued. Thank you very much. Wish you all the best. And you can enjoy these 3 days important symposia, and they have covered all important speakers. They are having a discussion on the cancer treatment, optimum care, then crystalline nanoparticles for the important platform for the drug carriers and drug development, then clinical research, how to promote, how to take care of the natural products as a base with respect to the drug delivery. So these are all the important areas covered, and Alzheimer's disease, oncology, important genetic diseases, and complex challenges, how we can take care during the drug development. These are the topics covered. I am really happy to congratulate the organizing committee under the supervision of Shivkumar ji for taking lead, and we are all with you to take care of the global health. Thank you very much.
Thank you, sir, for honoring us with your presence and for delivering such a memorable speech. We thank you once again for being here with us, for sharing your wisdom and for inspiring us all. Thank you, sir.
As we move towards the conclusion of this inaugural session, it is time for the vote of thanks. We now invite Professor G. Shivakumar, Principal, Geetham School of Pharmacy, to extend his gratitude to all the dignitaries, speakers, and participants. Please give him a big round of applause.
A very good morning to the dignitaries of the dais. It's my proud privilege to present in front of you to propose the vote of thanks for the Healthcare Conference 2025. At the outset, on the behalf of the organizing team, Healthcare Conference 2025, my heartfelt gratitude to all the individuals who are participating in this healthcare conference. I would like to thank our Geetham management for their unwavering support. Our President, Sri M. Bharat, a Member of Parliament, President of Geetham, for his unwavering support, giving us an opportunity to contribute and conduct such kind of healthcare conferences event. I would like to thank our Honorable Chancellor, Vice Chancellor, Pro-Vice Chancellors, Registrar, Directors, Deans, Heads of the Institute, Heads of the Department of the Geetham campuses across 3 states, Visakhapatnam, Hyderabad, and also Bangalore. I would like to thank, special thanks to our chief guests of today's function, Dr. A. Venkateswarlu sir and A. Ramkishan sir. Sir, we are blessed by your words, and we would like to thank you specially. Thanks goes to all the delegates, student participants, scholars, faculty members across the various institutions who have come to participate in this conference. Thanks extends to all the participants apart from coming from the various parts of the globe. Thanks goes to all the speakers, invited speakers who have traveled across the globe, national speakers who are here to participate, to contribute their expertise in this healthcare conference. Thanks to them. Thanks also extends to the local organizing team, the tireless efforts of the team, student volunteers who have made this event eventful today. Thanks extends basically to all the supporting staff, the hospitality team, AV team, the audiovisual team, print and electronic media. We are thankful covering for extending your support, and also to extend the facilities to sleepless nights to make this event very successful. Last but not the least, thanks to each one of the individuals who have been directly or indirectly involved to this event. So we hope this conference in the next 3 days will give us a very resourceful and a valuable input to the conference to all the delegates. And I would like to request all the delegates to actively participate, and thank you once again. Jai Hind. Thank you.
Thank you, sir, for your kind words.
As we conclude this inaugural session, let us now stand together in unity and respect for our nation. We kindly request everyone to rise for the national anthem as a mark for our collective honor and pride.
जनगण मन अधिनायक जय हे भारत भाग्य विधाता। पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा। विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा। तव शुभ नामे जागे तव शुभ आशीष मांगे। गाहे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता। जय हे जय हे जय हे जय 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 हे।
Thank you everyone. Please be seated as we proceed with the next segment of our program.
We now begin our first plenary talk by Professor Parimal Misra from Dr. Reddy's Institute of Life Sciences, Hyderabad. Professor Parimal Misra is a senior professor and chief scientist at Center for Innovation in Molecular and Pharmaceutical Sciences at Dr. Reddy's Institute of Life Sciences, Hyderabad. With over 28 years of experience in research, Dr. Misra has contributed extensively to the fields such as metabolic disorders, inflammation, tuberculosis, antimicrobial resistance, and genomic proteomics. He was also a visiting scientist at Feinberg School of Medicine, Northwestern University, Chicago, in the Department of Pathology.
Dr. Mishra has received numerous prestigious grants, including from Department of Biotechnology, DBT Center of Excellence, and Department of Science and Technology, as well as a collaborative RO1 grant from National Institute of Health, USA. He has been recognized internationally for his contributions, being elected as a Fellow of the Royal Society of Chemistry in 2016 and a Fellow of the Royal Society of Biology in 2017. In addition, Dr. Mishra was honored with the Chairman Excellence Award from the Dr. K. Anji Reddy in 2004. He is also a member of National Committee for the International Union of Physiological Sciences and the International Union of Pharmacology for the term 2014 to 2016.
Sir, please come on the stage. And now I request Dean Sir, Pro Vice Chancellor Sir, and Principal Sir to come on the stage and do the felicitations of Dr. Parimal Misra.
We are honored to have you with us, sir. Kindly enlighten us with your thoughts and insights. Over to you, sir.
Slide changer, can you give it to me? No, this is mine.
Hello, hello, is audible? No mobile phone. No, mobile phone. You can put it here. Yeah, I'm audible. Good morning, everybody, and all students, faculties, and there. And thank you for inviting Jagat, Pratik, and the students there. And almost I'm delighted. My teacher, mentor, Dr. Venkat, who recruited 1996. And the relation of what is the definition of a teacher? He's not the biological father, but when their student goes to teachers, he gives the second life. He has given life throughout India, so many students like us. So I'll just going to put a very important topic, and I want to tell you also, there is a reason behind to select the topic. Credit goes to Jagat, because when I last visited Visakhapatnam things, I'll come into their campus, beautiful campus. I saw something. End of the talk, I'll come into this to tell you. So if you see my talk, there are antimicrobial resistance, the 4 parts. Where from, whither to? Are you going to face another COVID-like pandemic soon? And that is a question mark. And we all together, all students and faculties, abroad scientists, we need to take a call which direction we are going. And Jagat told me, as is a plenary talk, I should not concentrate whatever the work we have done other than but give a overall things and what are the ground reality today in the world. On behalf of Royal Society of Chemistry and Royal Society of Biology, throughout the world and everywhere, we are trying to put this antimicrobial resistant menace and how to control it. And when we leave this auditorium today after this lecture, we need to try to execute today itself by setting it. That message I'm going to tell you what you're going throughout the world and several things and all. So now let me start. This is called a pandemic. And you know, it's a 2 axis. This axis is showing what are the year it has been done, just and there, and then how big it is. And if you see there, 200 million people died in the Black Death, bubonic plague, and then Spanish flu, though it's not originated in Spain, but it is name is Spanish flu, and then HIV and this thing and all. Then you come to the next. How many death happens? Forget about the years. Main of this, half of the year of gone during this Black Death, and then smallpox, then Spanish flu, and all those things happens up to COVID and these things. So there is a misconception, as it happens 1917, and COVID happens 2019 onwards. So pandemic will happen once in 100 years. It's wrong. It's not wrong, because until things comes very big, we don't care. So this is the questions we need to do that, what we will discuss today. And if you see this thing, I just put it into in these two categories. Before that invention of antibiotic and after the invention of the antibiotics, what happens to the human society? You saw that left page, this is a major outbreak throughout the history when that penicillin has not been invented. After that, when penicillin invented, there is an arm race going on with microbes with the human. Who is smarter? If he that smart, they are smarter. If they are smarter, we are smartest. But this arm race is going on. And see here, raising concern about future outbreaks without adequate treatment, how these things happen and all. Coupled with this, there is a major thing came. That is called the effect of climate change, melting glacier. Why we are so concerned? Because throughout the world, the temperature has been increased 1.5 degrees centigrade, which reached to the nodal point. Beyond that, if you go, it's a huge, huge thing. You all are scientists, you know how the microbes life cycle 16-minute replicates. If say 1 degree temperature happens more, how it will go more and all those. I'm coming into that. And because of the glacier change, what happens? New microbes, those who are frozen, it was coming out. And that's a dangerous thing. Like COVID, we don't know what could be the answer to treat these things. But this has been coming out and all. So today this is called antimicrobial resistance. And where is that Indian post-COVID situations? It is alarming. 30% neonatal death has been enhanced of this. You see here, this is COVID, post-COVID. This is neonatal sepsis. After this particular death of the kids, they are dying. It's happened 30% enhancement in this country. And then secondary infection after your COVID, it has enhanced due to death another 30%. So now this is in short what is called antimicrobial resistance. I'll not go for these details. And for what is the causes? Because of misuse of antibiotics, not taking care of so many things and adequate things. But most importantly, you have to understand 75% antibiotic used for agriculture farming. So regularity, Dr. Ramakrishnan here, 75% antibiotic used for the agriculture farming. And that is means the animal farming as well as this thing. And what is the danger lies, I'm going to come into there. Low dose of antibiotic is used there. And you see, this is the colistin resistance. E. coli is coming. And the most dangerous is Candida auris, which stays in the hospital setup. You can see the here for a period of long time, even in a month. That's why the ICU practice, Sir Ramakrishnaji, that's why all doctors, we are telling and talking, ICU practice needs to be changed. I'll come into that point. What could be the dangerous symptom, what we are having? And then challenges and initiative and the way forward. So many things. I'm not going to talk everything within 30 minutes of my time, 10 minutes question answer. I'll be telling what my teacher, Dr. Venkat, told me. Innovation, 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 and simplified innovation, and how to execute this innovation. That's why my talk, I'm going to talk now. Let's see how the antibacterial resistance from the farm to the table. You see this, where from it happens and how it comes, the resistance, spread, exposure and impact. And this is the dangerous things what we are telling, because of those who are from animal farming, they give low dose of antibiotic because growth of the animal, growth of the chicken, growth of other things. Dose is low, adequate. If you give more, that things will not be. That is such a greedy profit-making business happens. We need to talk, scientists, at a tune to stop this. Otherwise, it will be very difficult to contain. And you see the which part of the animal, here it is coming and which dose. And I'm giving you 70% of these are medically important, who are getting resistance. So it is a huge 360 problem, resistance, spread, exposure, impact is a total. What the counter OTC antibiotic is sold without prescription. I'm coming to that point. But this is the major things I'm telling you. We doctors, we scientists, we are politicians, we all have to come forward to take, but before do we have to understand what is the ground reality today. I'll give you some data and all those things I'm going to talk. Now let me, today as I told you, all these budding scientists are here and lots of faculties are from abroad. I came to learn with that exchanges, to learn things, to implement things and all. So what is this? What is known about antibiotic resistance? Then next I'll go slow to put the new third idea what we thought of. Is a primary infection, antibiotic, then the resistant antibiotic too, and let's see. And as a result, what happens? Those who are survival tactics, they become resistant. The MRSA stands for methicillin resistant staph aureus, one of the dangerous things what people can do it. So this is antibacterial resistance. This is very, very important part of this, any understanding antimicrobial resistance. First of all is survival of the fittest, then is the selection pressure. So you have to, antibiotic always comes under selection pressure. It should not cross the nodal point to do it. But if it is not that enough, I'll come into that what happens in the COVID time and what we are running through this danger point and all. So this is known about mechanism of antibiotic. It will not penetrate, or sometimes they are very, very smart, modified the target things, pumping out and out, a flux system, or inactivation, putting a phosphate group. Phosphate is a negative group. If you put in a thing, it's a completely different matters. These are the things known. But what is not known, that's why I'm coming to tell you and go slow from now onwards. So we all humans, right? And we have a puzzle. And how to solve this puzzle? Okay? How the innovation happens. Let us think, we human brain, how to do that. There is an idea. Then what we do? We challenge this idea. We got some alternative. And after that, provocation. And then we got out-of-box creativity. That is in-box creativity, stand of the box, then out-of-box. Like a mother bird, the way it teaches these younger things to fly, take you to the carnish of these things and give a push. From that, then started doing, right? But what is happening, if you see, this is a picture of a tadpole. If it is you signifies tadpole is a kind of a designed your thinking process, then it by chance, if it is designed, it will go, your thought process is like a tadpole. It can go this direction. So now, the point is change in thought process. If you tell what Dr. Ramakrishnaji is telling, we have to come together. If you think that I'm right, you're right, you have a confrontational thinking. Either you win or you lose, nothing there. Instead of that, let us think a parallel thinking. All thought process today's meeting put together. You see here is a circle and there is a contradiction, all are in sideways contradiction, not headways contradiction. As a result, what happens? We'll put this parallel thinking and constructive thinking. That is make a plan, then go, then go, then go. Like that we'll do it. And he saw that here. One second. There is a concept called SMART concept. Specific, measurable, achievable, realistic, time bound. This is called SMART. And then idea selection will be there, unmet medical need, input of clinician, number of prescription and all those, and you got that selected idea. All right? That is called design thinking instead of that. See, this slide came in 10 kilometers from here in the ICRISET campus. Dr. Venkat given a leadership thought. It's a very simple thing he told. There are three kinds of employees work in an industry. One makes it to happen, another watches to happen. Third kind, ask what happens. So he given us all those things, how make what happens to make it to happen. That is the thing through the constructive. I took it and extrapolated this thing to make it. Now this is human thinking. Next of my talk, don't underestimate microbes. Let us personify microbes next stop. Because after the CRISPR-Cas innovation, microbes has memory, right? So personified microbe. Don't tell it is a negligible small thing. If it is that small thing, then why the COVID happens? We can't tackle. So personified, take it is a miniature form of human. Not really, I'm just telling. Example, we need to solve a puzzle. What is that puzzle? Puzzle is to understand exactly how bacteria adapt their resistance. That means, you know, why it takes longer for some bacteria to develop antibiotic resistance and while others adapt very quickly? It happens, right? What is this puzzle? Why it happens? Some bacteria to take it, whatever the medicine you give it, MDR, XDR, but it takes time. Like you have a sepsis, you have to treat immediately, otherwise it will be organ failure, right? But it's one dose of IV you have to give it. But sticks is taking resistance, some are taking time, longer time. This is a new thought process coming. And here lies this main problem you have to understand. What happens? These bacterial microbes have an intrigon, kind of a gene system, toolbox. And when you have written something, there is a spelling mistake. That's called, you replace, it's called mutation. But if you have to change the entire system, then it will call as a insertion or deletion. That's you are changing your manuscript, right? So bacteria passes through the mechanism called recombinant, cut and paste. That means what needs to be done? If there is a hairpin, that is the DNA sequence, which is protruded from that finger, the 5' end, and you see this, all spacers, this is A, G, G, T, C, C, all are not same. And this recombinant binds to the DNA, some loosely, some very tightly. Let's see, you have to cut something. You have to hold tightly. It's like you have a knot, very tightly knot. You can cut it. If it is loosely knot, it fall again and again. It will not cut. It will not paste. Let us go together. It's the integrative science we are talking about today. How to do that? So this is hairpin sequence. So if you have complex, what I told, strongly bound, you can cut it and paste that. That bacteria does. If it is loosely bound, that you can't do it. And this is exactly the sequence, what I told you very lucidly, it has been done here. It is an cut and paste. That is the weakest point of a bacteria. Can we exploit that weakness of bacteria? Can we? If it is so, uncouple the cut and paste reaction. How to do that? You have a modern microscope. You have artificial intelligence technology. You can take this hairpin where it is binding. Can you have a customized antibiotics to do that, not to allow to bind? If it is bound, don't allow to paste. It's a cut and paste. That particular system to be unsmart. But bacteria is very, very smart. We need to be smarter. If they are smarter, we need to be smartest. But I'll give you some example later on what is happening. Let's see the COVID case. What happens? This is the COVID things given on my friend from Pasteur Institute of Paris. You know all those things. Well, what happens in the first phase of COVID or anything time? Infectivity rate was not that high, right? But the second phase infectivity rate was very high. Previously also COVID was there. What happens? Why the infectivity rate has been done and all? First of all, let me tell you. Sir, you have a house and your house is locked and Shrikanji, you have a key. What you do? You go there, your house, you take the lock, either your left hand or right hander, you put the key and the key has the groove. And then you put a positive or a negative direction. That means you give a trigger. Without that, just putting the key, it will not open, right? What is the trigger here? If it is there, next figure, See here, in this particular, this is bound to this particular protein, still it can't penetrate. Why? Why it has been penetrated? Because there is an enzyme called furin in the human blood. This furin, this cut it here, this particular, this sequence, what this people, COVID virus is happy. As a result, it's a continuous tunnel and virus is in. Where from it has been incorporated? Previously, if you see the Andhra Pradesh, Ramakrishnaji, you see there is a bird flu. After 10 chicken, after that 1,000, then lakhs chickens are dying. Very rapid infectivity rate. Because there is a protein called hemagglutinin. That sequence has been acquired by this COVID virus. Previously, it was not there. It's a polybasic. So, virus is smart. How they have acquired? What is the origin? Is it from human or is it from bird? Undersampling. We don't know. It may be still going on. Taking in 2013, when COVID came in Middle East, it was not that infective. Because of this particular furin cleavage, what I just told you, it makes all those things possible. So we don't know it is, so virus is very smart, very smarter. They know how to do that, spread it. How many still is under incubation, we don't know. This is the things what I just going to tell you here. And then, let's see, there is an emergency. Sure. When you have treated all those COVID people, because what is this clinical trial? We have to do solitary trial. You have a COVID, you need oxygen, or you alive or not with and without oxygen. And what are the drugs has been given? Several drugs has been given because we don't have a choice. Even pantoprazole has been given. Even aspirin, what Sir is telling, has been given. Even your atorvastatin has been given. Why not? Because this is the emergency. We have to safeguard. People are dying this thing. There is no other things to trade because it's unknown things. But as a result, how now we are talking about to exploit the weakness of these bacteria or microbes. Now I'm going to tell how microbes and bacteria exploit our weakness. What happens? Let's see. So all students, if you have any questions, you can do that. Let's see. There is a called inhibitor and activator. If you have to kill the bug, you have to kill 95% because it's a foreign body. If you have a diabetes, you can still slowly can do that, right? It is gained up a concept, you have to kill it. So it's very difficult, student, go to the 5th floor to 6th floor because you don't do exercise. But if you have to jump from 6th floor, under gravity it will be very quick. So it's a kind of a concept like that, but you have to kill the bug, right? You need IC90, MIC, not only broth culture OD, you have to play it on to see that any bacteria, microbes still alive or not. Otherwise you can't control the sepsis, right? And that is the difference between a chronic disease. I think many of you are here from cancer background and they are doing it. I'm a chemist, I'm a physicist and mathematics background, then switched to biology and SERB, and all of our colleagues made me a drug discovery scientist. So I taking this entire, all those holistic concept to pull all those things. So now the question is here, let's see. What is happening? There is a repurposing. One drug is going to be repurposed for the another. But when you do all biology or chemistry researcher, we put tag protein, flag tag and all. Is those protein exist in the human body? It is not there, right? It is artificially we putting to pull it down to see that what is happening for all those. So one drug is responsible for one disease. When you are repurposing for other disease, the criteria is changing. What is those criteria? I'm just telling you. A predicted model, let's see during COVID time what happens. They have only this IC50 data, EC50 data, not EC95, all right? They put it all those things, okay? And you see this as a result, what happens? Lots of drugs are on the basis of those data needs to be very much of positive, right? But it is on the basis of EC50. But you have to kill the drug, kill the bugs. It is EC90. What are those drugs? You see here. There are several drugs are there. But if you put EC50 and EC90, you see most of the drugs are getting out layer. It is not be active. What happens? We consumed all those drugs. We consumed all those drugs. And bacteria's microbes is very clever. They put this resistance. That's why post-COVID situation, antimicrobial resistance has come to that stage. Because we are given so many drugs, that time emergency. We don't know, Ramakrishnaji. We need to give. We have to drive the life of a people. But as a result, what happens? Now this is the period, coupled with your climate change, we are in a very, very dangerous situation. I'm coming to that place to tell you this thing. So now, there is another things what we are doing slowly under leadership of Dr. Venkat. So we need to do for this TB. MDR TB has gone up like anything. And because after COVID it's become very things. We work on a target called a charismatic mutation. Where from we got it? You may have seen this, our Golconda Fort. When Golconda Fort was attacked, what happens? That time, people to survive, they stop giving food and water to the enemies, right? Here, charismatic mutation takes the essential amino acids of a microbacteria. It is not present in human. So, taking this example, what you do that? If you can stop those essential amino acids to be made by inhibitor, will it be possible to kill the bug, not to harm other things and all? So, let me see what is happening. Here, this inside the macrophage, your latent TB, it stays and pH is 4.8. So, you have the drug to penetrate, it has to sustain this particular acidic condition. Otherwise, it will be several substrate modified, it will not work. Let's see here. This is MTB and this is, let's see, come, and this is charismat, and then it is make it prephenate and then essential amino acid, so microbacteria can go. If you have NC, let's go gun, binding to the NC, inhibition of charismatic mutation, and as a result what happens? As a result, deprivation of essential amino acids. So recently, what happened? This is the treatment regime for multiple drug resistant Ramakrishnaji that put by the government of India. Previously it was here. So this, it's not working or? Can you help me? Hello? Pointer is not working, can you? Okay, distance, now it is working? Or I have to come here? Okay, fine. So, let's see. So, this is the thing. Let's see, this is the TB therapy. What's going on? First oral line of therapy is these 4 drugs, right? This is country. And then you are putting rifampicin, and then what not? And third thing, it is showing that BPAL treatment. What you are suggesting, in addition to that, can you have a CM inhibitor? So the government thing is, instead of 24 months to 6 months, can you make it to 1 month? Here there is a lie. Linezolid is getting used. India is not faced to linezolid because of cost. Now linezolid is getting used. If linezolid got resistance, what we have? Another IV drugs, it both got resistance. Where you will go? So, this is the problem, what I just want to tell you. Now, as I promised, I will give you something for a primary invention, what Pennsylvania has done. In a CSIR journal in our country, there is a cartoon. One scientist got the best scientist award. And without any accident. So it was asked, what is the mystery? The scientist told, I can't tell loudly. You have to come very quickly to me. So ask a simple question. When will accident happen? If you work. If you don't work, there is no accident. This is negative. But if you don't allow microbes to come to your body, then the primary prevention. What Pennsylvania has done with the electricity, this is doctors. They are bringing all kids and teaching how to wash hand like COVID time. 20 seconds, if you wash your hand with sanitizer, if you have a tea, count 1, 2, 3, 4, 10, your lipid layer gone. Your DNA, RNA is there. If you don't allow to come, it will not infect you. I'm telling you, in Indian countries, in the village, urinary infection for the female is dangerous. Count is 10 to the power 14. Because in their country, because of their economic condition in the village, females can't use tissue paper after toilet. So you understand which direction we are going it. So this is my message to all of you. You have a cell phone, you have AI, you are stuck to it. Please like exercise fast, exercise second, exercise third. Do your wash hand fast, wash hand second, wash your hand until new drugs or some kind of a antimicrobial vaccine comes. So please student, you need to take a place today. Throughout the world we are going and telling, wash your hand, and wash your hand. And I'm thanks to the Jagat when he invited me to Gitom campus in Visakhapatnam. When he taking me for the lunch in the canteen, there is a lady stand and giving all those things to wash your hand, otherwise you can't take food. Thank you, Jagat. And that's why I put this title here, to do it and wash your hand and all those things to do that. And now, after washing this hand, those Pennsylvania, they are doing a quiz the competition of kids. You know, you are all from this country. You have a Nirma powder kind of a thing. 10 people going this ad, going Nirma wax to close your things. And that kid are very powerful. They're small but powerful in a positive way. Like bacteria is small but very negative. But small is beautiful, powerful. Nucleus is small, very concentrated, powerful. That put those kids and teach others. All we will educate today, all we will learn, and we will wash your hand. And all those things, I'm just telling what we are presenting and to given this lecture dedicated to Dr. Ranji Reddy and Dr. Venkat. He is the molecule man of our country, and Dr. Venkat, he put all those ragaglitazar, balaglitazan, all those fast Indian to put this drug in phase 3 clinical trials. So thank you, all of you, and thanks a lot. And I am looking for the question answer to learn. But wash your hand fast, wash your hand long, wash your hand like do exercise fast, exercise second, exercise third. Thank you a lot.
Thank you once again, Dr. Parimal Mishra, for your enlightening presentation. Now we would like to open the session for question and answers. If you have any questions, please raise your hand. We will bring the mic to you.
Any questions from audience? Sir.
Yes.
Okay, thank you. So good, sir, and thank you very much for the enlightened present. We really, got so much of information on that. And you have covered two aspects, which is alarming on that, which is the antimicrobial resistance, and the second one is the climate change. All right. So my question is like, how these two things are highly interrelated, whether the climate change is going to create more impact on the antimicrobial resistance, which we have been experiencing globally, So can you further give some insight into that?
It's a very important point. It may be endless talk, but I'm just telling you. First, when that temperature change 1.5 degrees centigrade, it caused the nodal point. And when the temperature increasing, your microbes replication time is changing very quickly. That's why 10 to the power 14 you are getting in the village during summer time for the infection and all. Second thing, as of glacier is melting, new things are coming out. Third point, what needs to be very, very cautious. In the ICU practice, when you are giving for the injection or IV, all those things, after that, are you taking this IC90 of the drug you are given or not? Other you are creating resistance there, because Candida auris is the next thing what is going to come big. Fourth is, when the hospital, all those things are coming out to the environment, are these totally, completely what is called pasteurized, or we are creating something there? All chemical industry, they are making for all those tons of drug for our life. How much carbon dioxide is coming for all those things now need to think of. These are all interconnected and giving a exploiting point for microbes for us to do this. If they are smart, we need to be smarter. But I have given an example. They are smarter, we are smartest. But we are smart, they are smarter through the COVID. So this arm race is going. Always take microbes as a miniature form of a human. Though they have a memory, not thinking that process, through the carton paste they can do that. So this is the coupling things happening. That's why antimicrobial resistance is a global thing. And I'm very happy to tell Dr. Ramakrishnan here, Telangana is the 7th state which has banned over-the-counter antibiotic selling. First is Kerala, who has done his 1,000 crores. Question is, antibiotic selling is good, but ban is good. What is the alternative? What is the alternative? Primary prevention, then permanent prevention and all those is coming. Azithromycin, I'm just telling, PPC it was 500 mg for 3 days. That with food. If you take anti-food, Dr. Venkat knows what taught us, it will be 4-time absorbance. Now the azithromycin is coming around more than 6 doses. And students and all, you just take cephalosporin like a murmura without doing. Students come and there you have fever, what happened? I've taken 2 cephalosporin. And this is the incubation period. All microbes are waiting to do more serious. And one thing, if you think I am an individual, I have a problem, this is a global problem. The resistance come to you, go to others, right? It is not an individual problem, right? The point is, we all use our AI, addicted to the AI, and that AI is reading your mind. They are telling that where you will do and all those. Put your AI in a positive way to do that. Don't spend so much of time on the cell phone. Please wash your hand and do the exercise. So then to some extent you can prevent yourself, right? Got my point? If you are not infected, you will not have a disease. It's a simple solution. I'm not telling the permanent solution. That's the throughout the world. I'm very thinking in one meeting, Royal Chemistry meeting, when I told this thing, people told, what is this in Hindi body bath? What is the big thing? I just showed one safety pin. I told safety pin is a very minute discovery. Can you within 10 minutes invent another safety pin and give it to us? Pin drop silence. Pin drop silence. Because COVID taught us washing hand, how can I do it? People thought it has alcohol. If you drink alcohol, then it will kill it. It will kill your cell layer there. It is on the palm. It's a completely different. Forget about the mechanism. Wash your hand and properly wash your hand. If you learn today, tell your kids. Your kid will tell them. Then all guardian will come. They will donate money to you. You don't have to spend anything. It's not awareness. If you like love humanity, if you like mankind, if we all want a better universe tomorrow, let us start from the small thing. What Dr. Venkat in his teaching always taught, start with simplified. When he took for lunch with Dr. Venkat, he told whatever today lunch, this is simplify. Simplify innovation. Simplify lunch, not complexity. It's very difficult to do what Tagore told, Rabindranath Tagore. To make it something simple to simplify is very difficult. That's why all scientists, I came to learn from all of you, including my teachers and all. Please give us feedback so that I can pass into the international community. I'm telling you, we are in a sleepless night. We are not understanding what next in our coming 6 to 8 months. I'm not telling. Now the point is, this question mark will be remain or not, all depend on us. It's a collective thought process. If you take a place, we'll rugate, we'll be smarter, then smart bacteria. If they're smarter, we'll be smartest. The tug of war will end. We can kill it. Like a clean toilet. That was the slogan of the country, right? Wash your hand. Wash your hand first, wash your hand second, wash your hand third. Do exercise first, exercise second, exercise third. You can prevent it. And prevention is better than cure. That's the point, what I want to tell you. If any other questions? Take the mic.
As we observing pandemic every 10 years once, so do you think, sir, in 2030 we can have another?
Well, this is a misconception. When AIDS came, 1980, right?
Yeah.
So there is no time gap.
Yeah.
Question is how you delaying it. Okay. The mother nature, it is a creation. We can't go beyond the natural law, right? Carbon given a biotechnology, silicon given your IT, information technology. Now this century has discovered that your virus or microbes has memory. This is one of the biggest discovery. So we need to stop that. We have to take into account and do that. Okay.
2030, sir. Do you think any?
Well, this all depends what AMR condition. Jagat knows, he's an expert. It's a very bad stage today. Okay. Let us stop it very quickly. Don't take antibiotic without showing doctors. Okay. All right. Okay. So that's my point and message to you.
Thank you.
Right. So any other questions? Yeah. Is that right? Yes. Thank you.
Sorry, Dr. Parimal Mishra, in the interest of time, we did not take any more questions.
Thank you, sir, for that insightful and thought-provoking address on the antimicrobial resistance. You have truly illuminated the gravity of the situation and the urgent need for collective action to combat antimicrobial resistance before it escalates into a crisis similar to COVID-19. On the behalf of healthcare team, I extend my sincere gratitude to you for sharing their expertise with us. I also thank all of your presence, your attention, and your commitment to this crucial cause. Let us continue this conversation beyond today and work together towards sustainable solutions. Once again, thank you so much, sir.
Now we request all the participants and esteemed guests to join us for the high tea at Shivaji Foyer. Now we will meet at sharp 12 noon for the next session. There is one more request. The participants of oral presentation, kindly submit your PPT presentation at registration desk during the high
Whoever
tea
has
session.
participated in oral presentation, kindly submit your presentation at registration desk.
After high tea, we will meet sharp at 12.
నెక్స్ట్ ప్రెజెంటేషన్ టాపిక్ ఉంటుంది కదా, ప్రెజెంటేషన్ టాపిక్.
Privileged to be here today, and I'm equally honored to co-host this event with you.
I hope you all had a refreshing break and enjoyed the high tea. It's wonderful to see everyone back and ready for the next exciting sessions.
As we continue, we have an incredible lineup of discussions, innovations, and insights that will shape the future of our industry. So let's dive back in with enthusiasm and curiosity. Thank you for being here, and let's make the rest of the conference just as engaging and inspiring.
It's my distinct honor and privilege to welcome a distinguished leader in the field of medicinal and organic chemistry, Dr. Srinivas Nanduri. Dr. Nanduri received his PhD in organic chemistry from Sri Satya Sai Institute of Higher Learning and has since built an illustrious career spanning both academia and pharmaceutical industry.
With over 3 decades of experience, he has made remarkable contributions to drug discovery, particularly in the areas of oncology and metabolic disorders. His journey has taken him through esteemed organizations such as Dr. Reddy's Laboratories, Biocon BMS Research Center, Origin Discovery Technologies, and Albany Molecular Research, where he has led and managed numerous innovative drug discovery programs.
His expertise in medicinal chemistry, drug design, and lead optimization has played a pivotal role in advancing pharmaceutical research. Currently serving as the Dean of NIPER Hyderabad, Dr. Nanduri brings a wealth of knowledge, leadership, and vision to the field of pharmaceutical education and research. Dr. Srinivas Nanduri will lead the proceedings as the chairperson for the first session. Ladies and gentlemen, please join me in extending a warm welcome to Dr. Srinivas Nanduri onto the stage. Now I would like to invite Professor Jagataran Das sir and Professor Shivakumar sir onto the stage to felicitate Dr. Srinivas Nanduri sir.
ఇప్పుడు ఆయన ఇంట్లో వినోద్ కుమార్ సార్ ఇంట్లో వచ్చాక ప్రొఫెసర్ వినోద్ కుమార్ సార్ నేను ఫెలిసిటేట్ అని చెప్పాను. మళ్ళీ ఒకసారి చెప్పండి.
Now Dr. Srinivas Nanduri sir will introduce the next speakers.
So invite him onto the stage. We'll tell you to felicitate. Yeah. Same happens to me. Sure.
So good morning and welcome to this exciting session, the first scientific session of the program. So the first speaker is Professor Vinod Kumar. He is a distinguished scientist and educator in the field of organic and medicinal chemistry, and he is currently serving as professor at the Central University of Punjab, and he is a PhD from IHBT Palampur, and he has made remarkable contributions to the drug discovery and green chemistry. His research area spans from CNS active compounds to opioid receptor ligands, with a focus on developing multi-targeting agents for neurological disorders. He has secured multiple prestigious research grants and received numerous awards for his contributions to science, having held prestigious academic positions including professor and head of the Department of Chemistry. His dedication to teaching, research, and administration is truly commendable. So with these few words, I would like to welcome Professor Vinod Kumar onto the stage.
We would like Dr. Srinivas Nanduri sir to felicitate Professor Vinod Kumar sir.
Hello. Hello. What do you? Hello. Yeah, yeah, that's fine. Okay. Thank you, sir. Thank you for the nice introduction. Uh, Respected Dean, Head of the Department, Professor Siva, Professor Jagtaran, distinguished speakers, and dear students. Thanks for inviting me here. I'm organic chemist and I'm trained as a medicinal chemist. My postdoctorate is in medicinal chemistry, and currently my area of interest is design and synthesis of multi-targeting agents, and we are targeting, our research group is targeting Alzheimer disease. So my talk is divided into two parts. First is introductions, means what we are doing, why we are doing, a little bit introduction about the disease we are working on, and second part I will be discussing the our molecules, what sort of molecules we are doing, and the in vitro and in vivo analysis we are doing. So I will be sharing some of my latest finding from my research group. So the Alzheimer. So for the young scientists, what exactly Alzheimer is? You might have watched Vicky Kausal's movie Udi, the surgical strike, and the mother of Vicky Kausal. So she was suffering with that disease, if you don't know it. So Alzheimer is basically characterized by number of symptoms: memory impairment, aggressive behavior, impairment in language, disorientations, and it's generally called as old age disease. So generally people of age more than 65 years, they suffer with it, and the stats, it shows the dangerous trends because there is increase in the diseases like stroke, heart disease, and HIV by 21%, 14 or 54%, but the deaths due to Alzheimer disease, they have increased by 89%. So this is a serious problem, and it was discovered in 1906 by Professor Alois Alzheimer. And the problem is, here you can see, it's a brain of healthy persons, and in the left side you can see the brain of a patient suffering with Alzheimer. So there is a shrinkage of the brain. The exact pathology of the disease is not yet known, but there are a number of factors which are considered responsible for the initiation and the progression of the disease, and these includes cholinesterases, monoamine oxidases, amyloid beta, metal ions, and lot many other factors are there, and the neurotransmitters which are affected. So there is low level of dopamine, serotonin, and acetylcholine. So this is the chemistry of the disease. So what exactly monoamine oxidase is doing? So here it is oxidizing the neurotransmitters, monoamine neurotransmitters, and in the process releasing hydrogen peroxide, which is further converting into hydroxy and reactive oxygen species. And these reactive oxygen species, basically these are responsible for the lipid and protein peroxidations, and finally causing the neuronal cell death. So this is the chemistry behind the disease. The other factor is beta amyloid. The problem is with the accumulations and clearance of the beta amyloid inside the brain. So it causes plaque formations, and these plaque formations, they resulted into formation of NFTs, and that finally resulted into the neuronal cell deaths. So lot many other factors are there which are associated with the progressions of the disease. These are the drugs which are FDA approved, and most of these drugs are targeting only acetylcholinesterase. And one of the drugs, this is memantine, is for NMDA receptors, and there are problems with the available therapies. So these drugs, they are causing a lot of problems: hepatotoxicity, bradycardia, abdominal pain, and because multiple factors are responsible for this disease. So targeting acetylcholinesterase only and inhibiting that esterase is not going to solve the problem. Okay, so there are many other factors that need to be taken into account. So, and secondly, these drugs, they are also developing drug resistance. So how basically, again for the young budding scientists sitting here, so how the drug discovery process, it works. So basically it focused on the drug which act against a specific target, and it's a sort of cause and effect relationships. And what we have to do, so we design molecules which can target the specific enzymes and stop its overexpressions. So and similar way, the single target, most of the drugs developed, they are single target based. So generally the target of clinical ripeness, that is identified, and then a best binder drugware molecules is used to be identified, and then the proof of principles that need to be generated, and through all those in vitro and in vivo assays, we need to identify a molecules of clinical relevance, and we got these drugs. We call it single targeting drugs. The main problem with the single targeting agents, one is they are ineffective for multifactorial diseases like cancer and neurological disorders, and secondly they are most prone to the drug resistance. Then what is the alternate, and what exactly multi-target directed ligand approach is? So in this case, because the diseases like cancer and neurological disorder, they involve this pathology, multiple pathology, multiple targets are responsible. So we need to identify a molecules which can simultaneously bind to the multiple targets. Then, so these multi-target activities, these are potentiated by either additively or synergistically. And then secondly, these multi-targeting drugs, these are comparatively less prone to the drug resistance. So why do we need it? We know that the cellular pathways, they are really very complex. So the single pathway is transport of these multiple steps and responsible for different physiological functions. So if a single targeting agent, if it stops one of the pathway, then that pathway can always adopt the alternative route, and the cause of the disease, or cannot be, the disease cannot be cured. So it is kind of a multi-locking system. If you are having multi-locking systems, so if we really want to open that lock, so we need to open all the locks simultaneously. Even a single lock, if it stays there, we can't open this. So these are the various strategies how we can synthesize multi-targeting agents. So these are different strategies which are being used. So and this is different, multi-targeting agents, they are different from the combination therapy, because in combination therapy we take different drugs for different targets, and this combination therapy has different issues, and the major issue is with the drug-drug interactions. So these drug-drug interactions, this is the major problem for the combination drugs. So that is why what we are really attempting is, so we need a master key, okay, which can open all the locks. But yeah, it's not that easy. It's easy to say, but it's very difficult to design a molecule which can bind to the targets of your choice and don't give off target activity. So that is major challenge. So how we can approach, or how we can focus on the molecules which are multi-targeting. So here, so the step-wise we can do. So first of all, we have to identify the targets, and then we have to design a molecules, and then we have to screen those molecules through the multiple targets, and then we have to come out with the best binders, so which can bind with the simultaneously with the target of our choice. Okay. And based on design, then we can go to the BAT lab design and check and screen the molecules through in vitro and in vivo assays. So these are the approaches how we can proceed. So first of all, we have to identify. So if we know that this particular pharmacophore is responsible for a particular activity. So for example, here you can see the molecules is active against acetylcholinesterase. At the same time, the molecule modulator, amyloid beta modulators. Okay, you can see. So we are having these molecules. So these molecules can be designed in different ways. We are approaching linkage strategy where you can link two different pharmacophore groups or two different units through a linkers, generally with the alkyl chains. And secondly, we are having fusion and incorporation strategies. So these are the strategies. And when we started in our research group, so we targeted acetylcholinesterase and monoamine oxidases. These were the two enzymes which we wanted to simultaneously target these enzymes, and at the same time, we were looking for a molecules which can show neuroprotective and neurorescue potentials. So these were the activities we were looking for. And when we have analyzed the literature, we have gone through the literature. So what we have found, so this first group of compounds, most of these compounds, they are acetylcholinesterase inhibitors. And what common pharmacophore features we have seen in these molecules is, so most of these molecules, they are containing the tertiary nitrogen group. And that tertiary nitrogen group, so it was easy to understand because acetylcholine is the substrate for acetylcholinesterase, and this group is already there. Okay. So we can easily link this molecule. It means, so this pharmacophore is required for acetylcholinesterase inhibitory activity. And by going through the literatures, we can looking at the MAO inhibitors. So what sort of the molecules which are inhibiting the MAO enzymes? So MAO A and MAO B. So we have again seen, so this was the proporzyl group. Okay, so it was, we found that it was binding with the FAD cofactor of monoamine oxidase B and giving the anti-MAO activity. So then based on this, so we have designed the molecules. So but we thought, let us have a heterocyclic moiety and let us attach the pharmacophore group, two pharmacophore group which are responsible for the acetylcholinesterase inhibitory activity and the monoamine oxidase inhibitory activities through any linkage, and then screen these compounds for the desired activities. So what sort of inhibitors or what sort of heterocyclic moiety we could have used? Okay, so we screened a number of heterocyclic moieties. We did the docking studies, molecular dockings against the crystal structures of these enzymes, and we found that the first one, the pyrimidine nucleus, so it was found equally potent both to the acetylcholinesterase as well as to the monoamine oxidases. So we decided, so first of all, let us start with the pyrimidines. So let us take pyrimidine as a heterocyclic nuclei and attach the two pharmacophore groups with that heterocyclic nuclei. So this was the pyrimidine nuclei we have started with, and the two pharmacophore features, as I told you, one of is with the tertiary nitrogen atom, which we are expecting to be responsible for the cholinesterase inhibitory activity, and the second one is a proprasyl group. Because so we came out with scheme, and then we synthesized a number of molecules with this scheme, and then we have screened these molecules. So these molecules were screened. So almost around 20 molecules were synthesized, and we also checked this intermediate, because this chalcone was the intermediate 3A to 3C. So here at the bottom you can see. So we have also checked the activity of whether this intermediate is also having some sort of activities. But here you can see it lacks the acetylcholine inhibitory activities. It has shown a little bit monoamine oxidase and monoamine oxidase B activities. But fortunately we could able to identify 3 potent compounds. So BB1, BB3 and BB8. And these compounds, these have shown MAO inhibitory activities as well as acetylcholine esterase inhibitory activity. So here we focused on the compounds not which were most potent against individual acetylcholine esterase or against monoamine oxidase. So we focused on the molecules which were showing balanced activity profile against both the enzymes. So these molecules, these were found to be active against these enzymes, and then we went for the in vitro screening of these compounds. So the first we did the reversibility studies. So the drug molecules, they need to be reversible so that the enzyme, it cannot be permanently damaged. So these molecules, they were found to be reversible against monoamine oxidase as well as against acetylcholine esterases. Then we did the ROS production inhibition activities. So the compounds, they were found to be effective against these 6-hydroxy dopamine induced neurotoxicity. We did the cytotoxic. We don't want these molecules to be cytotoxic, and these molecules were found to be safe even at the concentration of 25 micromolar. We did the kinetic studies to know the mode of action of these compounds. Again, we did the docking studies to again look into the mechanisms, or to look into the what sort of enzymes they are interacting at the active site. The molecular dynamics studies, they were also performed to see the thermal stability of the molecules inside the cavity, this active cavity. And so these were the first series of compounds, and then taking leads from this series. So we thought, okay, because I have shown you that in the first series, the pyrimidine nucleus, the molecules which were devoid of pyrimidine nucleus, they were found to be active against the acetylcholine, inactive against the acetylcholine esterase. So what we thought, let us go with the diproporzyl. So let us put these diproporzyl substituents, and because from the previous study we realized that this pyrimidine, because this nitrogen atom, tertiary nitrogen atom presented the pyrimidine nuclei. So it is giving us the anti-acetylcholine esterase activity. So we synthesized another series of compounds where we are having this diproporzyl substituent. So again, the number of molecules were synthesized, and we got again some of the lead molecules. We did the in vitro and in vivo. I'm not going into those details. So, but again, from taking again leads from these studies, so we decided to go for because in the initial study we synthesized O-proporzylated derivatives. Second, we thought let us go with the N-proporzylated derivatives, and what is their impact, how they are impacting the activity. So in the next study, so we have synthesized these different derivatives of a series of N-proporzylated derivatives by keeping other rings and other features constant. So in this study too, again we could able to identify 4 leads, and these molecules were found to be very, very active. So here you can see at BP14 and BP15, the acetylcholine esterase. So these activities are in nanomolar range. Okay. So these compounds were found to be highly potent against acetylcholinesterase in nanomolar range and showing activity comparable to the standard inhibitors. So other assays, cytotoxicity, reversibility assays, which I have discussed. So these studies were performed. Unfortunately, this series of compounds were found to be irreversible. And N-proporzyl group is basically responsible for the irreversibility. But in the other kinetic and ROC and production inhibition studies, these compounds, they were found to be showing good inhibitions. And then we did in vivo. So we did it on the mice models. And first of all, we checked the blood-brain permeability, blood-brain barrier permeability, and the molecules, they were found to cross the blood-brain barrier. And in the locomotor studies and other behavioral studies, in the Alzheimer model of mice, so these molecules were found to be quite effective. And keeping in view the high activity of these molecules, again, the docking studies and the stability studies through the molecular dynamics, so these studies were conducted. So because the results were really very interesting and encouraging, so we went with the patent. We got this patent in 2022 for these molecules. And so from here, so we again synthesized few more series of compounds which were active. So we did some other modifications to find the leads. For example, we put this phenyl nucleus here, we synthesized another series, and we got some leads. So, and to carry forward these studies, what we thought, so because we have changed, so we have done a lot of changes with the pharmacophores, with the alkyl chain and with the proporzyl chains. We have extended the chain length from proporzyl to pentyne chain. Okay. And then we want to carry forward. We want to see the effect of the heterocyclic nuclei. The other heterocyclic means what the other heterocyclic nuclei they were going to do. And then we came out with this chromone nuclei. So this chromone nuclei, again, in the literature, it was found to be effective against acetylcholinesterase and monomer oxidase. So we thought, let us take chromone nuclei as a heterocyclic nuclei and let us put the pharmacophoric groups attach this pharmacophoric group and screen these compounds, these molecules. Okay, so again, we did the studies. We went with the synthesis of different derivatives, putting different substituents. So first of all, we checked the inhibitory activities, and in addition to acetylcholinesterase and monoamine oxidase, so we have again added one more target, that is beta amyloid, which was again found to be responsible. So we screened these molecules, because in the initial studies at a maximum concentration of 20 micromolar, so the percentage inhibitions was calculated and the IC50 values were calculated for the active or for the compound of interest, which have shown multipotent profiles. So those compounds were identified, and we have find out the IC50 values for these compounds. And those molecules which were found to be effective against acetylcholinesterase and monoamine oxidases. Okay, so we screened these molecules for the beta amyloid aggregation inhibitory activities. And here you can see, so this is the control. So if you compare the control with the test drug, so you can see there is a visible change in the aggregation inhibitions of beta amyloid. So these molecules, these have shown aggregation inhibition up to 58%. Okay, and the resveratrol, standard resveratrol. So beta amyloid inhibitory activity more than 50% is considered as very good. And these molecules were found to be really very good inhibitors of beta amyloid. So these have shown again the druggable characteristics. Further cytotoxic studies, molecules were found to be safe. In the reversibility studies against acetylcholinesterase and monoamine oxidase B, these molecules were found to be reversible. These were found to be again neuroprotective and shown displayed this ROS inhibition potentials. So because these molecules were good, so we went with the in vivo studies. So this time we went with the zebrafish model of Alzheimer. So it was a scopolamine induced Alzheimer's, and the these geographies they were treated with these test compounds, and the results they have shown the effectiveness of the drug molecules, and these have shown potentials. So these molecules were found to be effective. Okay, and further, so these were different tests in vivo analysis we have conducted, and the molecules of our interest they were found to be effective. Okay. So the computational studies, dynamic studies, okay, and again, so taking leads from these studies, so we have synthesized another series of compounds by now introducing the proporzyl group, because in the initial study we didn't have the proporzyl group, so we introduced here the proporzyl group, and again synthesized another series of compounds, and again these compounds were evaluated against all the three targets: acetylcholinesterase, monoamine oxidase B, and beta amyloid. And again, so these molecules were found to be showing good inhibitory activities against all the targets, and the druggability were again. So we have screened the molecules for other activities which I have discussed with you, and so this is the latest work. It's unpublished work. We haven't published it yet. The studies are almost completed, and we are communicating it to GMedCam. So again, in this molecule, in this case, now we have extended. We extended the chain attached to the chromone, because initially there was only the phenyl ring was directly attached to the chromone nucleus. So here we have introduced another alkene, a olefinic bond here. So it is just to facilitate, because we are having acetylcholinesterase enzyme which is having extended cavity with Cas and Pas, and the molecules they are supposed to interact with both the sides. So and as per our expectations, the molecules they have shown very good anti-acetylcholinesterase inhibitory activities. So most of the molecules, so they have shown good inhibitory activities, and these molecules were further. So some of them even 94 up to 94%. So the IC50 values were calculated, and then we again, so we did the other studies: beta amyloid inhibitory activities, reversible inhibition activities. These were compound, again these compounds were found to be little bit irreversible. They have shown partial inhibition potential. And the computational studies also, we have conducted a computational study, and this is the inferences we have drawn from these studies. So actually, because we did structure activity relationship studies, and we have screened, or we have put various substituents, and we have realized that this N-alkyl chain is specific. For example, for pyrimidines, so the morpholine nuclei was found to be, and the para-substituent morpholine nuclei was found to be most effective. But in this case, it is inhibiting the activity. So it was specific, means there is role of the heterocyclic ring we are selecting, or we are working on. So these are the conclusions. So I have got the list. I have to conclude. So I thank you for your patience learning, and I must thank all the credit goes to this team. So I'm just here the face of this group, but the credit goes to these young scientists. So Binay, Piyush, Vijay, Naveen, Kailash, and this face, I think most of you are familiar with. So we are having Ashish and Bharti. So Ashish is a wonderful scientist. He's a very good worker. He did a lot of chemistry, organic chemistry there. So he was always there with a lot of good ideas, and yeah, he's good. He's good in chemistry. He has only one limitation, because when you see samosas, so he's always uncontrollable. He likes samosas too much. So that is only limitation he's having. And I thanks the funding agencies, UGC, DST, CSR, and MOE stars for the generous funding. So thank you very much. Yeah, I think I finished within
Thank you very much, Professor Vinod. It's very interesting. Actually, when most of the medicinal chemists think that selectivity is what is very important, it reduces a lot of toxicity. I think you have shown beautifully that, I think multi-targeting is a approach, particularly for the CNS. I think, it works very well for the CNS, but I'm not sure about the oncology area. Maybe in the kinase, again, still we look for selectivity. But it's very interesting talk. I think we can take few questions from the students, from the audience. Anyone? So please raise their hand so that we can pass the mic.
Okay, I assume I was too good to explain everything. Yeah.
Okay.
Hi, Doctor. So this was interesting to listen to you because A beta for
Yeah, you can.
Everybody is looking for A beta 42 aggregation, inhibition, so many things. But how to go forward now? Next step. Because in India, it's very difficult to identify where, if in vivo you have to go, where is the model? So there we always stuck. So have you identified anything for future?
For in vivo?
Yeah.
But we are working with, we are having collaborations with the pharmacologists. So we are working on that. So yeah, it's difficult.
Biggest hurdle. In India, nobody is having in vivo model for Alzheimer. Especially A beta 42, if you have to show that this is inhibition is happening.
Yeah, that's true.
But it's very, very good to see that so many molecules you have identified and then they are working against the inhibition as well as other receptors also. Thank you.
Thank you.
Good afternoon,
Yeah.
Good afternoon, Dr. Vinod Kumar. So really, your lecture was very inspiring. I am a fellow of Royal Society of Chemistry, but today the pharmacy students lacking chemistry are not that much deep into chemistry. They are not going. That's why your lecture they are not coming and asking questions. But you don't mind, without chemistry, pharmacy is not there. So all these medicinal chemistry, drug design, all aspects, whatever you are given, like pharmacophore, how the multi-targets, all these inputs, definitely all the students will take into it. But they are unable to come up with the questions. The reason is because it's very much specifically to the CNS. Those who are working on the drug design only can come up. So, but it was really very much thought-provoking for those who are working in the medicinal chemistry. Well, sir, well done. Your lecture was very nice.
Thank you very much.
Professor Vinod, actually, I think I have one question. So, the first series you mentioned that it is a irreversible inhibitor, right? So, because of the propargyl moiety. So, what are the alternatives you are thinking? Are you thinking of changing that propargyl to some other things?
Yeah, actually, most of the ligands which are targeting monoamine oxidases. So, if you are having a propargyl, and we have seen in the literature that if it is N-propargylated, if you are having N group there, so almost all, around 99%, they are irreversible in nature.
Okay.
So, only alternative is because the O alkylated, they are found to be equally effective. So, yeah, we can shift. We can, if you are doing O alkylation in spite of N alkylations, so that will work for that.
Okay.
Yeah.
So, with this, I think, thank you very much, sir, for a very exciting talk and very informative talk. It's very, thank you very much once again.
Thank you very much. Okay.
Sir, can chromonuclear markers be used for early diagnosis or prognosis for Alzheimer's?
It's very difficult. Actually, the main problem is the symptoms we are having for this disease. So these are very normal symptoms, like normal memory impairment. You forget many things. Means it's very normal. It means many of the people they tend to forget the things, but it's very difficult, and unfortunately, when it is at the last stage, so when it got severe, only then usually it could be diagnosed. So that's the unfortunate part of with this disease.
So now we move from medicinal chemistry, we move to the computational chemistry and artificial intelligence. So I have the pleasure of inviting Dr. Yurisha Novak, a distinguished researcher in the area of computational chemistry and artificial intelligence. He is a PhD from the University of Zagreb, Croatia, and he has contributed to leading institutions including Ruđer Bošković Institute and the University of Rijeka. His work spans artificial intelligence, cybersecurity, and molecular modeling, and earning him multiple awards and international recognition. So with these few words, I would like to invite Professor Yurisha to the stage.
We would like Dr. Srinivas Nanduri sir to felicitate Professor Yurisha Novak sir.
This one or no? Cell phone is not there. No.
Okay. Hello to everyone. To everybody. My name is Yurica Novak. I'm from small country Croatia, from the middle of the Europe, more than 6,000 kilometers from here. First, I would like to thank organizers from bringing me here, for invitation, for giving me opportunity to present my recent work to you, to your students, and hopefully to initiate some collaborative projects. Okay. We have some special letters in Croatian alphabet, so that's why the name of my institute is misspelled. It is Ruđer Bošković Institute. Ruđer Bošković was one of Croatian physicists. Okay, so my idea is to speak today about computational aided drug design, and first I want to show you a few cases where I used it in collaboration with my colleagues experimentally. So first, you can apply it in cancer research. This is case with BRAF inhibitor, where Pratik designed different inhibitors. My job was to run molecular dynamic simulations to see how they bind, if the complexes are stable through time, and if there is any possibility to improve those molecules. Second case is in antiviral
drug design. I don't have to tell you anything about COVID. I will just say that we were working with the main protease of SARS-CoV-2 virus. It is a very good target protein because it is responsible for cleavage of the polyprotein that is synthesized, and without it, you don't have major viral particles. Our idea was not to design a competitive inhibitor, but we we have actually different approach. We wanted to design allosteric inhibitors. The thing is that what we wanted to prevent it to make a dimer, and if it is in monomer, it is not active. Third example, it is concerned with post-traumatic stress disorder, because my colleagues, experimentalists, they noticed that if they treat rats who were exposed to big stress with compound resveratrol, they found it that the cortisol levels were lower, and the question was why. So my part of the job was to do again molecular dynamic simulation to see if it can bound to that protein and somehow inhibit its initial role. The most part of my talk will be about two viruses. They are completely different from the perspective of a pharma company. The first one is monkey fever virus. It is specific for some regions here in India. The other one is HIV. So for KFD, let's first define the research objectives. So we know what we knew actually was that it is also inactive unless NS2B cofactor is bound to NS3 protease. So we want to again design allosteric inhibitors, and first we have to identify allosteric binding site. So short background, as I said, it is a flavivirus family virus. It has 3 structural proteins, 7 non-structural proteins. We are interested in NS3 protease. This NS3 protein has multiple functions, one of which is being protease. And without, as I said, this short 40 amino acid peptide cofactor, when it is bound, then he has his function. From a pharma company, it is not interesting because yearly there are below 1,000 new cases, but as a virus, it is interesting to scientists. Here is short workflow, how we approach the problem. So we didn't have 3D structure. We use AlphaFold to design NS3 protease and also to design NS2B NS3 complex. With those structures, we enter into molecular dynamic simulations to see how actually they behave through time. And then first part was to do analysis. We actually wanted to see why is this NS2B factor so important. Once we know its role, then we went into this other branch. We did cluster analysis. We identified relevant conformations of that protein. We searched for allosteric binding sites, and then we did classical drug repurposing study where we were searching for potential allosteric inhibitors. Okay, so here are results of this first part where we were simulating bare NS3 protease and NS2B NS3 complex. We showed that both complex and only NS3 are stable through time, but there are some differences. And okay, you can see, but these A and B graphs, they have different scale. The main point is here that there are much more fluctuations in bare NS3 protease compared to NS2B, NS3 complex. And this is also confirmed in this subgraph C, where radius of gyration was calculated through the time. The most important part is shown in D graph, where root mean square fluctuation is presented for both systems. These metrics show us how flexible each residue in the protein or in the complex is. And we can see that when NS2B cofactor is bound to NS3 protease, its flexibility is decreased. So the other point is that, as you can see here, those residues that are responsible for catalytic activity, their flexibility is barely changed. So the main changes are outside binding pocket. We did free energy calculations, then we did decomposition of free energy. This is important to see which residues contribute the most to the binding. So these are just numbers. On these figures, they are depicted so we can clearly see where those residues are. So now, next step was to search for allosteric binding pocket, but for this we wanted to see which conformations we have. Why this is important? Because if you have, let's say, different conformation of one target, of one protein, the binding pocket can have completely different geometry. And if you want to have effective drug, you have to effectively target both those conformations. So we identified two dominant conformations, both for NS3 and NS2B, NS3 complex, and those dominant conformations differ if you have complex and if you have bare protein protease. We also perform principal component analysis. Why? Because we wanted to see which molecular movement are responsible for this effect. And what we see, unfortunately, this is just a static picture, but you have those arrows showing the biggest movement. In bare NS3 protease, the main part is here, this loop that is moving. And in NS2B, NS3 complex, we don't have this movement. So the thing is that this movement is actually blocked by NS3 cofactor. So this was just supporting our initial hypothesis that we could actually target allosteric binding place. And we identified two binding places. As you see, their geometry parameters are different. So when we did drug repurposing study, we did docking to both conformations, to conformations A, to conformations B, because, as I said, we want effective drug that is binding to all relevant conformations of our target. Here is just a list of our hit molecules, and for top 4 molecules, we performed molecular dynamic simulations. Why? We wanted to see if this binding really has some influence on dynamics, on the stability of the complex. And our finding was that, as you can see, the allosteric binding pocket is far from active site. Those catalytic residues are presented in yellow. And the biggest conformational change, it is those two areas that are encircled. So what we noticed is that we have great geometrical change far from the binding site. And those geometrical changes completely changes the geometry of the catalytic pocket. So we have this, I would say, even large amplitude motion, because if we measure the distance from, let's say, this loop to the relevant active site amino acid, we have distance from 8 at the beginning of the trajectory of the simulation to more than 20 angstroms. That means now we have a lot, we actually open this target, and now the solvent can easily enter the active site. Okay, so completely to summarize, we see that cofactor has structural effect on NS3, so it minimizes the fluctuations. We identify two relevant conformations. We identify allosteric binding pocket, and we propose several already known drugs that could target those allosteric binding sites. The other virus is HIV. We wanted to design QSAR models that will accurately predict biological activity. We want to design, we wanted to design new anti-HIV molecules. Again, just as I said, for monkey fever virus, it's not interesting to pharma. HIV is much more interested. We have several dozens of already approved targets. The main problem with HIV that it has a high mutation rate, from 10 to 100,000 more mutations per genome compared to human cells. And here are just the different targets of HIV cycles that are targeted. I think 5 years ago, more than 25 million people were receiving highly active antiretroviral therapy. So it's a therapy where 3 different drugs are targeting at least 2 different mechanisms. Okay, now how we wanted, how we planned and how we, let's say, solved our problem. So first we can divide actually to 2 lines. First line is this QSAR modeling, where we have to collect the data when we have to design QSAR models that will then be used in this second line, where we will actually design new anti-HIV drugs. Okay, so we actually have the 2 databases. One is for anti-HIV data, where we collected effective concentrations needed to observe protective effects in infected cells. We selected 1,000 molecules classified as confirmed inactive, and we have 200 molecules classified as confirmed active. And we also, for cytotoxicity, we used concentrations needed to inhibit the growth of the uninfected cells, or IC50 values, and here we took only 100 most toxic and 100 least toxic molecules. Okay, so we actually did QSAR modeling. I don't know if you have any idea why is QSAR so hard problem. The thing is that space, this chemical space, is enormous, and we are actually trying to use some specific descriptors to filter those molecules. I can do analogy. For example, imagine you are on a cricket is popular in India, right? So imagine you are on a world championship of cricket between India and Pakistan, and you want to find Pratik. You know only that he's 175 centimeters high and his shoe number is 42. What is the chance that you will find Pratik if you don't know how he looks, if you know only his height and shoe number? Chances are small. And what if I show you the picture of Pratik? Will that increase the let's say, successful rate. Will you find Pratik more easier? Yes, you will. Why? Because not only you see his face, but you also see relative, let's say, orientation of his eyes, nose, mouth. So the same we should apply in a chemistry. The thing is that 3D structures are very important, and that's why we use this 3D QSAR model, a Cinderella shoe. So it is a pseudo-receptor model where you just overlap, superimpose 3D structures of molecules, and then you recreate your target protein, or it doesn't have to be protein, it has to be just target. The other way around is this continual molecular interior algorithms that we also used. Here is also another analogy. Imagine you have, I don't know, a plate made of glass and made of steel. They look, let's say, the same. They are hard, for example. But what if you take hammer and try to crash it? That from glass will crash immediately, while this steel one will not. So here the point is that you cannot say everything from just watching it. You have to know something about its inner property. And that's why we use this common algorithm, because it actually reflects this. So the thing is that, again, let's go into the chemistry. Some molecule, how they will act when they have some inner, let's say, force. Will they rotate? How easily they will rotate? So this is everything important, because you know, there is this induced fit model in enzyme kinetics. So these are very important things that we also have to include. So we were combining those two modules, then we applied multiple linear regression, we applied neural network to design QSAR models. We have this desirability function. Thanks. We have this desirability function that was actually used in QSAR modeling. We designed 18 anti-HIV models. We designed 12 cytotoxicity models. These are just part of the results. Now, once we have efficient models that could predict biological activity, that could predict toxicity, now the next step is drug design. So what we did, we collected from this anti-HIV dataset molecules that are the most active. We search for which these molecular moieties are the most abundant, and we took them. And we actually say, okay, let's try to do modification on these 20 molecular structures. We will introduce up to 3 different substituents that are here with this star. Okay, we had 6 different models how we can do these substitutions. This is just one of them, where we have this GF3. This is the main fragment with 3 open valencies. And now, using your chemical intuition, you have to just put some different fragments to close all open valencies and generate chemically correct structure. Okay, so here is just one example how you can do it with these triazine molecules. So we have different fragments. We generate open valencies by removing, in this case, only hydrogen atoms. Then we combine them, and we have new molecule. In the process of drug design, we used the genetic algorithm when we were generating new molecules. Why? Because we want to optimize. We want that our molecule is highly active and less toxic. And in this genetic algorithm, our chromosome has, in this example, 1, 2, 3, 4, 5 genes. How many fragments you have, that many genes you have. Each fragment in its own library has its unique number. And these are just here. So in parent molecule, we have 5 genes. 2 parent molecule can produce 2 child molecules using crossovers, and then each child can experience point mutations. And again, this formula is not well displayed. Here is our main result. So after 10,000 steps of optimization, what we got? We got molecules that are not cytotoxic and that experience very good anti-HIV activity. And here I just inserted the initial graph. So this initial graph, this upper part, shows initial let's say condition, because initially we randomly selected 100, we randomly designed 100 molecules. And as you can see, those molecules were both very toxic and barely has some anti-HIV activity. So during this 10,000 generation, what we show? We show that we obtain very active molecules with, that are barely toxic, and another property of this genetic algorithm is that he filtered out those fragments, those main block fragments that are either toxic or don't have good enough anti-HIV activity. Okay, so these are our top molecules that we designed. Then we compare how similar those newly designed molecules are to already known anti-HIV drugs. The highest, we calculated Tanimoto coefficient, it doesn't matter, so you see that it is quite low. 0.24 is the highest. That means that our molecules are completely new. And then we tried to do, actually we were interested by which mechanism these molecules work. So we did molecular docking study for protease and integrase, I think. And actually the main conclusion is that we cannot just based on docking say by which molecule, by which mechanism these molecules work. Besides this, we successfully developed and validated QSAR models to predict both anti-HIV activity and cytotoxicity. We implemented the whole framework, the whole workflow, how to design new molecules based on fragments you have. And yes, mechanism of action remains an open question. I would like to thank Pratik, Shivananda, Vladimir and Maria, funding, and of course I have to acknowledge for SUPEC, supercomputer, for having actually access to this supercomputer. And I would like to thank you all not to fall asleep during computational chemistry talk. Thank you.
So thank you very much. Now I think we can take a few questions. Yeah, please go ahead. I think please keep the mic.
Sir, you spoke about non-structural protein earlier. So when you're talking about the non-structural protein as the target molecule, as your target, are you talking about only using it as the for that particular virus, or can it be extrapolated for other viruses, because non-structural can be a functionality which is across the viruses? So will it work with the theoretical AI models or quasar models, or what in that capacity?
I think that those models can be transferred to other. For example, this first virus, monkey fever virus, is a flavivirus, and The closest one is Zika virus.
Okay.
It has, when comparing just this NS3 protease, similarity is 50%.
Okay.
With active pocket.
So because the action is similar, we can extrapolate it to other viruses as well and try.
You can do it, but you cannot say that it will 100% work.
No,
because
not
also for this.
No, the principle, I mean.
Yeah.
In principle, yes.
Yes,
Right.
yes. Yes,
Thank
you
you.
can. Welcome.
How do you select your protein? There are many different protein is there in PDB data bank. So how do you select that particular protein for that you're talking or computational work? Can you?
Well, for example, for this HIV, you have to know, for example, viral cycles, how he multiplies. And then you have to target those proteins that are critical for his function. For example, let me just go back to the HIV. Thanks. Oops. May I have my presentation, please? Okay, so the thing is, for example, in HIV, it is RNA virus. And RNA has to be translated to DNA. And this is the critical enzyme that is doing this. It's perfect target. Because in human cells, you don't have analogous function that will translate RNA to DNA. So you can target this one. Protease, for example, it is critical because the virus synthesizes this polyprotein chain, and then it has to be cleaved. And HIV protease cleaves this protein, polyprotein. And without this step, again, you will not get functional virus. The thing is that, as I said, you have to know why you pick that target.
Okay. How do you think about isotopic labeled APIs? Few they are in the market. So in docking studies, how it would be like very useful?
Well, in docking studies, I don't care which isotope is.
Like proton or like C13 or F19?
In docking, you just have to define which atom you have.
Okay. How do you think about activity?
But I think that this is more important in experimental validation.
I see.
In these parts of drug design,
okay,
I'm not interested in, I just take the most abundant and that's it. But again, if you know the mechanism,
okay,
and if you want to, let's say, do some quantum studies, then it would be important, yes.
Thank you.
Okay.
It is not a question, rather just to make your thing more interesting. You initially told that this CAD is attractive to academicians but less attractive to the industries. So if you can give one or two examples to our students where CAD has helped a molecule to come to the market. If you know one or two examples.
They are.
If you can tell something to the students, they will be interested in this.
But unfortunately, I'm bad with history.
Okay, thank you.
But they are. But I know that also one of HIV drug was designed in that way, because they know the active site, as I said, and they know that there is one critical water molecule inside, and then they design a molecule that will not allow that critical water molecule to enter the active site. And without this water molecule, you actually broke the mechanism. So it is important.
Hello, am I audible? I think To add up to your answer to this question about the targets, in the life cycle of HIV, the targets start from the attaching to the receptor, maybe CD4 receptor, then followed by reverse transcriptase, protease, then integrase. All these enzymes, various targets are taken. From those, the proteins are selected. Then particular protein is isolated, and then a target docking has been done. Thank you.
Sir, most of viruses are following -1 or any frame shifting, ribosomal pausing process. So why we are selecting particular proteins only? We can select ribosomal, or we can target only ribosomal frame shifting. If we will select ribosomal frame shifting, then we can overcome the multiple protein synthesis process.
It depends on the researcher which target you will take and to which approach you prefer. But I agree, it's a good approach. Yes. But also then if you want to apply machine learning models, for example, you have to have some experimental data. So then the question is, are there any experimental data concerned with this mechanism?
So thank you, Dr. Rish. I think it's a very exciting talk. Particularly you were the first KFDV, where you have done the allosteric inhibitors, non-competitive, and also the second one where you have tried to address the mutations, especially which are rapidly evolving with the HIV. I think the work is very, very interesting. I think as Jagat is saying, that a lot of complementarity is there both in academia and industry. I think all of us should be collaborating with you. I think so for the chemists, to make the molecules, and particularly, so why you chose this? Have you compared with, because you are doing the allosteric and non-competitive? So have you done anything with the competitive inhibitors also? Compared it, the first one.
If I did what?
No, the KMDV, when you have done this allosteric inhibitors, yes. Have you also worked on the competitive inhibitors?
Yes, we did. We did also drug repurposing study, but the thing with these flavivirus proteases, they are charged, and the geometry is quite flat. So it is hard to design a specific inhibitor of this protease. So that's why we tried to design this allosteric.
Okay.
It is possible, we did it, but somehow for this type of protease, I would prefer allosteric. Yes.
Okay. So thank you very much. I think it's a very wonderful talk. Thank you very much.
Thank you.
Thank you to Dr. Srinivas Nanduri sir for taking the lead as the chairperson. Attention all the participants, the oral presentations will be conducted simultaneously in rooms D104 and J211, and the poster presentations will be done in Block E, first floor and second floor beside Office of Geetham School of Pharmacy. We kindly request all the presenters and volunteers to proceed to the designated rooms promptly. Volunteers will be available in the area to assist if you need. Thank you for your cooperation. Lunch will be served at Shivaji Foyer, and we kindly request everyone to proceed there as the break will start from now till 2:20. And please make sure to return to this auditorium as we have an exciting session lined up afterwards at 2:20. Enjoy your meal, and we will look forward to seeing you back. Thank you. We hope you had a pleasant lunch break. Let us now resume with our program with the next session. It is with great honor and deep respect that we welcome an eminent scientist, educator, and visionary in the field of organic and medicinal chemistry, Professor Jagadharan Das, Dean of Pharmacy Geetham, Visakhapatnam, Andhra Pradesh.
With over 25 years of experience spanning both academia and corporate world, Professor Das has made invaluable contributions to the field of drug discovery. He earned his PhD in chemistry from the prestigious Indian Institute of Science, Bangalore, and further honed his expertise through postdoctoral research at esteemed institutions in France, Germany, and Japan.
His leadership roles as Director of Research at Dr. Reddy's Laboratories and Vice President of Drug Discovery Research at Panacea Biotech have been instrumental in advancing pharmaceutical innovations.
His dedications to academia is equally commendable, having served as a professor at Shulini University, Baddi University, and a guest faculty at Naipur Mohali before joining Geetham.
His research in molecular design, synthesis, and SAR studies continues to shape the future of medicinal chemistry. Professor Jagadharan Das sir will lead the proceedings as the chairperson for this session.
Ladies and gentlemen, let us extend our highest regards and a warm welcome to Professor Jagadharan Das onto the stage.
Good afternoon, everyone. I think after lunch, the attendance is very thin. I hope the people will join very soon. But anyway, to keep our program going on time, I just introduce the next speaker, Professor Raj Kumar. So I know Professor Raj Kumar just after his completion of master's degree from Naipur Mohali. He is also incidentally from Dr. Reddy's, ex-Dr. Reddy's, alumni of Dr. Reddy's. So he worked with one of my colleagues at Dr. Reddy's after his master's degree, and then he went back to Naipur to complete his PhD degree. He is a distinguished academic and researcher, and his contributions to the field of pharmaceutical sciences, particularly in medicinal chemistry, have earned him recognition as one of the top international scientists, and he has been featured in top 2% scientists by Stanford University. With over 15 years of teaching and 21 years of research experience, he made significant strides in cancer research and drug discovery, developing innovative therapeutic agents that have the potential to transform medical treatments. His passion for both scientific discovery and mentorship has shaped many bright minds in the field. Dr. Kumar's expertise, especially in the areas such as anti-cancer research, green chemistry, and the development of novel chemical entities, is widely respected. His dedication extends beyond the laboratory, having served as head of the department and dean shaping the future of pharmaceutical education. Please join me in welcoming Dr. Raj Kumar to the stage. Dr. Raj Kumar, please.
We would like Jagadharan Das sir to felicitate Professor Raj Kumar sir.
10 minutes.
Yeah. This is also working. Is it open? Good afternoon. Am I audible? Yeah. First of all, I would like to thank the organizer, Professor Jagatram Das sir, Dean, Head of the Department of Geetam, and all the dignitaries, and the Vice Chancellor, Chancellor of this university. I can see that there are very few students, but anyways, I'll try to deliver the things what I can do. My area of research is anti-cancer research. So in anti-cancer research, by seeing the kind of group we have, I will make it very simple. From last 15 years, I'm working on cancer, and I can see that no major breakthroughs have come. However, the methodology as well as the modalities for treating the cancer is changed, is it drastically improved, the patient-driven way it is. And if you talk about cancer, I did my postdoc on cancer therapeutics, but when I came back to India, I went to some private college for teaching, and then I joined Central University of Punjab, Bathinda. Bathinda is a place where I came to know that there are many cancer patients are there. So we tried to find out why and what is the reason. So there are multiple things, and there was a train going from Bathinda to Bikaner, taking the patients from Bathinda to Bikaner. Bikaner is in, I guess, Rajasthan. So that triggered me that why it is so, why there are so many cases. So there are multiple groups in our university like environmental science, chemistry, and other departments which started working on cancer, what are the reasons. So we as a pharmacy people, we worked on cancer therapeutics. We have another department, human genetics and molecular medicine. They try to collect the blood samples from the villages, nearby villages. Mostly in villages the cancer patients were very, number was more, breast and other cancers. So then they found, they took the blood samples, they identified the important markers, and it happened so even soil sample, pesticide use, arsenic, so many things contributed synergistically in the cancer patient's occurrence. Anyhow, it is mostly in the villages. Then AIMS came into the Bathinda, so they also have a separate cancer division. So because as a medicinal chemist, what we can contribute, we can think of new modalities, new drugs, treatments, regime for the cancer, as cancer therapeutics. Today I am just showing you one of the small story or one of the research paper I am going to discuss with you, that what we have done, my team has done. The title is quinoline-bridged hydroxamates, design, synthesis and anti-cancer. This looks like a medicinal chemistry kind of thing. Let's start. If we talk about cancer statistics, so it is predicted that by 2030, the developed countries will have 47% of patients will not survive, and 66% of cancer patients will not survive in developing countries. The reason that developed countries still have all the facilities and all the things available to them, and developing countries still struggling. That's why we need some other treatment options, and you will see that these cancers are most commonly occurring. However, I have not covered other pancreatic and other deadly cancers as well, but these are the most prominent cancers. I want to draw your attention to this data. This what we did in it is 5 years data. We are compiling more 5-year data. We published in JMedChem, and we found out that if you dig into the FDA approved drugs during that 5 years, 2015 to 2020, the search for, the number of drugs approved for the cancer therapeutics is still, in the majority. The reason that it is a dreadful disease and it is a multifactorial complex disease. So the treatment is very tricky and you need a polypharmacology kind of a cocktail to be given to the cancer patient. And you can see the other diseases as well, the ranking. So this also indicated that the research in the cancer is demanding that you need to develop more therapeutics. Not only immunotherapeutics, small molecules, monoclonal antibodies, as well as degraders are now coming into the market. They are in the clinical trials, particularly for the cancer. So I have shifted my area from small molecule to degrader recently, but in this presentation, I will focus on small molecules. This that difference between the normal and the cancer cells, we generally say that it is proliferating cells, fast dividing cells, and we can demonstrate with the help of some cartoon that the normal cells die by cellular suicide. This is called apoptosis when they behave abnormally. What happens when the cancer cells can avoid apoptosis and survive by producing pro-survival protein shields? It is a very notorious cell. You will see there is a network of cell signaling pathways, and cancer cells bypasses many of the pathways and does crosstalks between the signals. And cancer cells can also survive by making defective or mutated death proteins or not making them at all. This is the slide actually borrowed from Hanahan paper, which depicts that there are many hallmarks of cancer. So I'm telling you that you cannot develop a single molecule to hit the cancer, but there are many mechanisms, like these are the some important characteristic of a cancer cells evading, it becomes kind of immortal, and many growth factors are overexpressed, nuclear factors are overexpressed. This is uncontrolled division of the cells, and many pathways are being abruptly disturbed, and many of the things are downregulated or upregulated. So this complex, you can see a network. This is just a small thing, but there are many such complex mechanisms which take part in proliferation, cell growth, protein synthesis, and EMT metastasis, angiogenesis. There are so many factors involved. Think about in normal cells, everything is under control, but in cancer cells, anything can be disrupted. And if cancer cells bypasses one of the, if some wiring is rewired, then anything can happen. And this gene expression can be, so cancer cells divide so quickly that they have less time for gene expression and don't mature properly. And things are not under control, it is beyond the control. And there are many proteins we discussed, people have used many ways to combat cancers. Some are working on protein kinases. Morning we were discussing about protein kinases, threonine kinases, serine kinases, tyrosine kinases. Okay, and classically, if you talk about cancer therapy, still chemotherapy is being used on the forefront, then radio and palliative, many methods are there. If you talk about, so basically topoisomerases and histone, they make a condensed matter of the chromatin. So if you look at this, the chromosome and the histones are being wrapped by the DNA, and the relaxation of the DNA depends on the histone as well as the next sequence, that is topoisomerases. So if you see that, and mostly histones has lysine and arginine which are charged, so DNA is negatively charged, so compactness of the DNA on the histone depends on the acetylation and deacetylation of the histone protein. So if DNA, so the idea is that DNA should not relax, it should not make further DNA so that we can target the cancer. And if you see this slide, the DNA is wrapped around the histone, and histone takes the help of enzyme HDAX, which generally does the deacetylation. So if we inhibit the deacetylation, the intactness, compactness will be there. DNA will not, relax. And if it doesn't relax, then what happens? It will not go for the further DNA synthesis, and hence the protein synthesis will also not take place. The second event is when it relaxes, there is some supercoiling, and topoisomerase 1 or 2, they come and they relieve the tension and make it relaxed, so that they allow it to further undergo the transcription and other post-translational, protein synthesis and other things. So this is, if we inhibit HDAC, then the next event, so there is a resistance mechanism. It will be good idea if you inhibit both HDAC as well as topoisomerase, and further selectivity is also required. There is challenge, a lot of challenges are there. HDACs, there are 4 classes of HDACs. Which one to inhibit, which one not to inhibit. Topoisomerase, there are 1 or 2. And in bacteria, topoisomerase 4 and gyrase are also there. So people on anti-infective group, they are also working on that. So this was the idea. So if you look at topoisomerase, there are 2 kinds of topoisomerase, 1 and 2. One cuts one strand at a time, and 2 cuts 2 strands at a time. So this is the only difference. And one is protein ATP dependent, other is not. So this is just a cartoon to show that how your topoisomerase, then, and how topoisomerase inhibitors act. For the students who are in B.Pharm, they might be, studying some fluoroquinolone, antibiotics and some other things, anti-cancer chapter is also there. So some are, act as poisons, some act as the catalytic inhibitors. So if they act as poison, then there is a ternary complex, they just stabilize the ternary complex. But when they inhibit the ATP binding site of the topoisomerase, then they act as a catalytic inhibitor. Of course, they are reversible inhibitors. So these are some of the, I have already informed you that 5-fluorouracil and these methotrexate, they as a anti-metabolite, and these therapies, folate antagonists, they're still in the market, they are being used, and people are mixing them with folate inhibitor, EGFR inhibitor, folate inhibitor or other inhibitors. A cocktail they are just trying to, or some alkylating agents as well. So the targeted anti-cancer therapies have revolutionized the cancer therapeutic regime. Actually, starting from the imatinib, which was first, tyrosine kinase inhibitor, and then people are now working on. Further, in kinase also, so much resistance, mutation has come, selectivity is the issue, and in kinase also, now people are targeting EGFR KRAS, EGFR multikinase, because morning it was asked that in protein kinase resistance, yes, resistance is there, and dual or multi-inhibitor approach is still being used. Lapatinib is one of the examples. So these are the, gefitinib also has resistance. People are trying to make some dual inhibitor, but major are in clinical trial, but effectiveness is still awaited. And some side effect, cardiotoxicity, these are the major problems with all the, anti-cancer therapy. So what is the role of small molecules in targeted cancer therapy? This is again a 5 years before data, and we are compiling another 5 years data. We'll publish in some good journal. Let's see. So this also shows that mostly nitrogen heterocycles, they are still having the majority if we talk about the small molecule. We are not talking about here monoclonal antibodies or degraders, but still degraders also contain nitrogen heterocycles as a core molecule. So still FDA approved have a nitrogen heterocycles, but nitrogen heterocycles still have some problem, because as a heterocycles they also become susceptible to aldehyde oxidase. So what is the problem with the reported topoisomerase inhibitors? Everybody knows that camptothecin has a E-ring, and natural product people are sitting here, so camptothecin is derived from the natural product, and it has an E-ring, and E-ring is susceptible to the hydrolysis, under alkaline hydrolysis I would say, and people have tried to change it. Mark Cushman and Pomier group at Purdue, they were trying hard to develop more and more topoisomerase inhibitors and TDP1 or 2 inhibitors, where they have replaced this skeleton with some other heterocyclic moieties where this E-ring is not at all there. But still, camptothecin is not FDA approved, but this topotecan is still FDA approved, but it also undergoes the hydrolysis. It is also not that much stable as the other drugs. Plasma stability is still low, and these are some of the known camptothecin derivatives which are also FDA approved. Doxorubicin, which is antibiotic, also it is being still used. So these are again these are some of the problems with the existing drugs also. Always there is, as a medicinal chemist, we have a tendency, or we have to discover new molecules devoid of cardiotoxicity, because topoisomerase is a cardiotoxic problem, should have selectivity, selective profiling, and less toxicity to the normal cells. So if you look at topo 1 and topo, remember I told that topoisomerase also has 2 types of enzyme, H-topo1, topo 2, and H-topo2 is also again of 2 types, alpha and beta, and beta is not good, but alpha, beta, if you inhibit beta, then cardiotoxicity will come. So alpha, inhibition alpha is good. In H-topo1, 2 also, if you inhibit one enzyme, other will, compensate it. So again, resistance issue. So this cancer cell is very smart. It is notorious and it compensates the things and starts synthesizing the DNA. So this is the way the elevation of the H-topo isoform 1, inhibition of one, if you inhibit one, other will, increase. So what could be the strategies as a medicinal chemist? So as a medicinal chemist, either we should change and we should go for the combination therapies, which people are doing, but it's not a medicinal chemist. People, pharmacologists and doctors, they are using combination therapies, just mixing 2-3 drugs, polypharmacology kind of thing. Second is new chemical scaffold, because if we see, for example, in EGFR inhibitors, if one framework is, has been used like gefitinib, quinazoline, and now the enzyme knows, okay, gefitinib has come, it will develop mutation, point mutation. So threonine to methionine. So gatekeeper residue is changed, and now the gefitinib cannot enter inside the cavity, ATP binding site. So this kind of thing we can develop, but how long we will develop, I don't know, that new chemical scaffold. Second is, as we discussed, hallmarks of cancer. They are really good. There are many hallmarks of cancer proposed by Hanahan, where you can target not only the receptors or targets which I have shown, but also microenvironment like pH and viscosity of the cancer cells, pH of the cancer cells. People are working nano delivery, like because depending on the pH, esterase cleavage and other things, pro-drug approach, many approaches have come, like targeting. Some people are utilizing now reductases, because the cancer cells has more ROS, more reductases. So if you develop a drug which can selectively go to the tumor which is hypoxic in nature, oxygen is less, and it will get reduced and get accumulated and toxic to that particular cancer cell. And multi-target approach, as Professor Vinod also discussed in case of Alzheimer, people are also using multi-target approach, and I will be discussing this multi-target approach, that if we can develop topo 1, topo 2 and some HDACs simultaneously, we may achieve some selectivity. Okay, so this data I will show up to the in vivo level, but yes, it needs further. I will tell the disadvantage and drawbacks also, so that we can take some message out of it, okay, what need to be done further. So these are some of the reported topoisomerase and HDAC inhibitors. Yes, there are some other, again, they are not in the market, they are in the discovery stage. People are working on the selectivity and other things. And our approach, like, it is a classical approach. This is not it is a well known that you have to have a cap, and it is my experience that if you want to develop topoisomerase 1 inhibitor, the molecule should be flat, because it works with DNA. And if you want to develop topo 2, it can have a chiral center or some, conformationals or some bridged or something like that. But if you can have something, if you can develop at least more topo 1, less topo 2, it's fine. Dual inhibitory property will suffice the work. So you can have a cap of that one linked with the linker, and then at the end you have a hydroxamate moiety which can act as a zinc binding domain, in particularly in HDAC 1 and 2 classes, where have zinc as a cofactor. Recently people are now avoiding this hydroxamate also. People are working on now benzamide kind of moiety. Benzamide, they think that they are safe, because hydroxamate also have some other issues. But anyways, this work was done 2 years before and published recently. I cannot change it now. But I have to tell you as a medicinal chemist that there are things, what are the advancements in the field. So based on this, if you see that quinoline is a common factor, now people may ask, quinoline is a well-known drug, right? Quinoline, why people are, but if you talk about quinolone, which has a double bond and NH, and it is a ciprofloxacin, they are well-known antibacterial, but work by DNA gyrase, gyrase and topoisomerase, that are also topoisomerase enzymes. So this is also having properties. And then we can link it with the linker, and simultaneously we can inhibit HDAC and topoisomerase. But what is the proof of concept? Seriously this works? So what we did, so this should be the methodology actually, that synthesis, structure elucidation and all these things. This is a journal slide for all the students, that how we go for medicinal chemistry program. This could be the approach, One thing is missing in this. I would like to, at the end I will ask you, what is missing in this? What should be the approach? So this is the whole summary, that this is the approach of a medicinal chemist. But there are some points missing which you need to, as a student or a participant, you need to answer what is missing in this. Okay, we will come back at the end. So we developed this molecule and we synthesized this molecule. We published ACS omega, chemistry part we published, but we were interested how this molecule is, going to. So we have a confocal microscope and we have a, we want to see where it is going. So if it is showing red fluorescence and it is after 12 hours, see the nucleus, this is a nucleus, zoom nucleus, the nucleus is disintegrating. That means this compound is going to the nucleus and finally after 24 hours there is a disintegration. So nucleus is finally the cell death is taking place and all these things. But really it is happening, so in nucleus there are so many enzymes. So we tested this compound on topoisomerase 1, 2, HDAC it was not showing activity because there is no chain, and we were also seeing that okay whether this can act on tubulin, tubulin is also there, but it is not acting on tubulin. So okay, this was the preliminary results, so we thought why not to take such kind of template and link it with a linker and put a HDAC probe. So we synthesized very few molecules, not hundreds or these molecules, no. We just did some, we got some result and we wanted to see the proof of concept. So we developed some molecules and we synthesized this ester and then followed by the, what is this, hydroxymination, and we got the hydroxamate esters, sorry. And these are some of the molecules. 1B is already there, this you can see 1B, and then these are the molecules. We were having trimethoxy, 2-methoxy, we wanted to see whether it is showing the activity or not. Then we realized that first we conducted the topoisomerase 2 and 1 assay, topoisomerase 2 decatenation assay. It is being, we developed in our lab, it is being done on agarose gel, and we found out that particularly this 5C compound was, decreasing the level of kDNA. So that is good, that means if kDNA level is high, then the relaxation will be high. So it was not allowing it to relax. And then 5C was also, if you find out the IC50, you will surprise that in the above concentration it is 100 micromolar. Referee asked us that why you are using 100 micromolar. Yes, this topoisomerase we need 100 micromolar. It doesn't come in nanomolar or something like, because it is in abundance. So the IC50 is very high, in particular in this case. And we calculated it was 67.76. So what? The next point was whether these compounds have some effect on topo 1 as well, because topo 1 is also participating in causing knitting and relaxation. So we found out that yes, 5C is also showing, 5C was also, the densitometric plot you can see that relative concentration of supercoiled. More is the amount of supercoiled DNA, less is the relaxation because supercoiled. And so you can see that our compound, our compound is inducing, it is not, it is actually during our compound, the relative concentration of supercoiled DNA is high as compared to topotecan, which is FDA approved drug. So that means that in our compound's case, the supercoiled DNA level is high, that means relaxation is not taking place. Okay. Then we wanted to see, because it has a tail and then hydroxamate, so we wanted to see whether it is inhibiting some HDAC or not. We did some, at the time we were having only 3 HDACs. These are a really costly affair. HDAC kits are very costly and you have to prepare some cell lysate, nuclear extract, HeLa cells we take, and then so many things we have to do. We carried out this reaction and we found out that 5C is particularly inhibiting the HDAC at IC50 11.94. So it was pretty selective as compared to the other compounds. We submitted this paper to JMedChem, then the Stuart Kohn, he asked, no, you have to give all the HDAC profiling, then only we will be convinced. So we carried out particularly one compound, the 5C, we did all HDAC profiling, and still our compound was, so we purchased kits and we did all the things, and then still it was showing good activity against HDAC 1 only. So some like SAHA, it is a very known, vorinostat is a very well known drug, which is a pan-HDAC inhibitor. So it's okay that sometimes they want selectivity. So our compound was HDAC 1 selective. And we did some, our collaborator actually did, Eric from Sweden and his postdoc Vibhuja from, now he is in Bradford. So both of them they carried out, because there were 2 compounds, 5C and 5G. Anti-cancer effect was there. Both have one, the 5C is having 1-methoxy and 5G is having 3-methoxy. And they wanted to see, and there was one carbon extra in 5G, how they are binding. So we found out that 5C and 5G, they have a different binding recognition pattern. One is the zinc binding site is, the inverse binding was there with respect to this thing. But of course, we could not answer with the help of biology, right? The crystal structure we need to have. So anyhow, JMed Chem editors wrote, no problem. we can accept your proposal that the binding mode is different. Okay. So I will wind up, sorry. So we did molecular modeling and H topo 1, topo 2 to prove, okay, how the binding interaction, why 5C is coming out to the best. So that's what we wanted to support. Then we develop, this is HDAC 1, HDAC 6. Yeah. Then we saw their effect on the cancer cells. We found out 3-4 compounds, 5B, 5C, F, they were showing good activity on A549 cell lines. 5C was the best, and in 5C also we checked, this was the A549. But if you see this H1975, this is a mutant lung cancer cell line. So Gefitinib was not effective, Saha was not effective, etoposide was not effective in that. So our compound was having less than 250 nanomolar concentration, nanomolar IC50, particularly in case of resistant lung cancer cell line. Then we did some assay on 3D also, it was performing well. We chose other compounds also. In 5C, you can see that the 3D culture behaves like in vivo. So we developed 3D model, because earlier it was 2D model, no 3D model. And you can see the density of the cells in, in the inhibition of this thing in 5C. Then we did some other couple of assay, because what is happening when we are giving treatment to the cancer cells? What is happening with respect to the redox parameter, what is happening with respect to the mitochondria, what is happening with respect to the apoptosis, whether cell cycle is being arrested, at what stage, particularly they were arresting at G1 and G2M phase particularly. So this is all data, I am summarizing it. We did J1 assay, and ROS, 2 compounds were increasing the ROS. So ROS act as a double-edged sword in case of cancer. If you have a higher ROS, then only also it can be thing. So once you are targeting topoisomerase, HDAC, then subsequent other incidents are also happening after this, post effects. Okay, so mitochondrial defenseless mitochondria is there. Now it can happen because ROS level is increased. So there are so many things happening. So cell death, mode of cell death, we compared with other inhibitors, and we checked the Western blotting also, and we saw that PCNA level was PCN level was decreased. So PCN level should decrease in case of the anti-cancer effects. So it is numerous processes you can see. I have summarized because time is less. So these are the events happened which we did, and we also carried out in vivo assessment also. This was carried out at Naipur Mohali with Professor Tikku. So he is expert in all these 3D in vivo experiments, and 2 doses it was done. Subcutaneous injection was given. And we told it as a 15 mg is a low dose and high dose is 30 mg, but yeah, so this was the protocol followed, and the outcome was that 5C in a high dose, means 30 mg per kg, was effective, and it was able to reduce the tumor weight as well as this you can see the size of the tumor also, like Saha treated FC low dose and FC high dose was pretty good, 30 mg per kg. And we did some then tissue, we removed the tissue, and we did Western on that also, that what happened to the level markers. And we did some qPCR also, and histone like H&E staining. And what is the problem now with this compound? Everything is going good, The problem is that when we submitted, the problem was with DMPK actually. When this Arajan researcher, because we have shared the paper with them, so they carried out DMPK studies, this in vitro and in vitro clearance studies. In clearance studies, it was having high clearance. So the problem is, you can see that where the drugs fail. So now we are working on the lead optimization of the quinoline scaffold where the clearance is high. So it was when we saw that in vitro DMPK, the clearance was high and the plasma stability is less. So means the time should be high, and some cytochrome P enzymes, they are involved in the metabolism, and it is inhibiting CYP1A2 also. And these were the predicted, not predicted, these were the metabolites obtained, and mostly you will see that the gluconide conjugation, sulfate conjugation, and all these things. So we have to really look at the soft center of the quinoline as well as the linker where we can control. So we are working again, and somehow let's see where we can go further. So these are the problems associated, like as a medicinal chemist, when you go, you should also look for the in vitro DMPK profile, and then first do pharmacokinetics, decide the dose, then you go for the in vivo. So that is the take home message. So this is the whole story, and we have published this work in JMedChem. Thank you so much for listening.
Thank you, Dr. Raj, for this nice presentation. Now we can take a few questions. Particularly, I think students, please come forward and ask any silly question.
They have understood everything.
You see, I have seen Dr. Raj growing from very early stage and starting as a chemist at Dr. Reddy's, and now he has become a professor. And from his presentation, I can see that how he started simply with starting the cancer therapy and then gone to the drug discovery, which is a very complex process. So,
hello,
yeah,
sir.
yeah,
I have a question to Dr. Raj. It's a great work.
Where
first
are
of
you?
all. I'm here, this side. To your right side? Yes. So my question is, did you get any chance to do PKPD in vivo?
Sorry?
Did you get a chance to test the PKPD parameters of your drug?
No,
in
no,
vivo?
that's what I'm telling. PKPD, actually we submitted, and in vitro DMPK we came to know that it is showing
having high clearance. That
So
we got
it is
it.
understood.
Right.
And so that's why we didn't go, and we have to modify the lead now.
Right.
And keeping the efficacy constant.
Right. So, but you didn't do
No,
any
no, no,
of
we are now modifying it.
Okay.
But yes, but I would say I have my another work is going on with Michael at Germany, in Germany, Hanover Medical College. From last 2 years I submitted them compounds, and last 2 years they did in vitro, but they said then we sent them the good amount of compound for in vivo. I was hoping I will get in vivo results. They said no, we are optimizing, we are first checking the stability of your compound in the plasma. So, so that girl, that student, she is withdrawing the samples after every 2 hours, but it is difficult to test because after withdrawing a sample also drawing a sample is not a blood sample, and she showed me the graph where some of the compounds are not stable, but some compounds are. So she will take those compounds which are active as well as they are surviving in the plasma, and then they will look, first they will complete PKPD and dose, and then they will go for the in vivo, which we are doing wrongly actually in India. We just publish initial results with in vitro or some in vivo. That is a problem. But I am thankful that JMedChem accepted it, but if referees would have been very strong, then they would have opposed. It took 1 year to publish this work.
Oh. Thank you, sir. Thank you.
Okay. Any other question?
So everything is understood. That means if there are no more questions, actually just now I was telling that I have seen him grown from chemist to professor level, and the way he has started his topic and way he has explained, I feel that he is not only good scientist, he is a very good teacher as well. So let us thank him for his nice presentation, and I think students, you must have understood that discovering the molecule is not the ultimate. That is the first step. And see that he has found out one molecule which is active at the 30 mg per kg. You just think about the 60 kilogram human being, what is the dose it is required. So he has lot of work to do, and he has already found out the loopholes in the molecules. He has to modify the molecule. I wish him all the best, and let us thank for his nice presentation.
Thank you.
So coming to the next presenter of this session, it's my great pleasure to welcome Dr. Tyagarajan Madhesaran. Dr. Madhesaran is an associate professor at the IMU University School of Pharmacy, Kuala Lumpur, Malaysia, where he has dedicated his career to advancing the field of pharmaceutical sciences. With a remarkable 6 years of academic research experience, Dr. Madhesaran is a recognized expert in the design and development of mono-olein-based liquid crystalline nanoparticles for topical and targeted drug delivery systems. His deep expertise, combined with a passion for teaching and mentoring students, has made him a respected figure not only in the classroom but also in the broader scientific community. His work continues to push the boundaries of innovation in drug delivery technologies, and we are honored to have him share his insights with us today. Please join me in giving Dr. Madhesaran a warm round of applause as he takes the stage to enlighten us with his invaluable knowledge. Dr. Madhesaran.
We would like to invite Professor Jagataran Das sir to felicitate Dr. Tyagarajan Madheswaran sir.
Hi. Is it audible? Yeah. All right. Okay. All right. Can I have the slide changer? Okay. So good afternoon, everyone, and myself, Tyagarajan, working as associate professor in International Medical University. First, I would like to thank Professor Dasa for a nice introduction, and next I would like to thank Professor Shiv Kumar for giving this excellent opportunity to interact with all the young minds here, and then I feel humble as well as honored to share the stage with all the eminent scientists in the great nation of our country. Right. So with that good note, let me start my presentations. I'm going to share some of the insight on the liquid crystalline nanoparticle, okay, which is a lipid-based nanoparticle, how it is useful, some of the insights on that particular carrier. So this carrier I have been working since 2010, where I started my PhD in South Korea, and that with this carrier, like I have been like working for almost like 14 years time period. And I'm going to give some insights on how this nanoparticle is useful. And I'm just connecting with the previous talk by Professor Rajkumar. Oh, that was a very comprehensive presentation on the discovery phase, where they have a very good, they do all the synthesis and come up with the product, and they have some limitations or challenges, particularly on the pharmacokinetic. All right. So we all know that there are certain limitations. I can skip these slides because it's a kind of like textbook explanation on that, and just I take from Professor Rajkumar where he has left that. the pharmacokinetic parameter. It could be any of the factor that could result in that pharmacokinetic limitations. So one of the things which I will be mainly focusing on this talk is about the solubility aspects of some of the anti-cancer drugs. Okay. Before that, I will give a very simple overview on some of the statistics on the nanomedicine. What is the current status? It's kind of a brief report card on the nanomedicine. Okay. So the nanomedicine is keep evolving. I don't want to define what is nanomedicine. By this time, you all know what is nanomedicine. Okay. All right. So the growth phase is on an average, it's at 12%, and Asia will be the biggest player in the coming 10 years, according to these statistics. Okay. All right. And 39% comes from the oncological disease. So there is a great potential in the market to capitalize on the nanomedicine based drug delivery system to overcome some of the limitations we have. Okay? And if you see, focus on the strength. So given the opportunity as a pharmacist or a chemist, we need to have a clear understanding on the physicochemical property of the drug and how we relate with the clinical demands, basically, right? So there, the drug delivery will have a huge role in overcoming all those limitations. Okay, all right. So this slide is like the milestone of the nanoparticle-based drug delivery system in the market. It's what to highlight. In the early 1994 or early 1990s, we have the first product in the market, and this field is evolving. And recently, during the pandemic, we had the Pfizer vaccine or Moderna vaccine, which basically shield the mRNA or make more stable dosage form or vaccine, okay, in which it preserves the mRNA stability outside the body and once it is administered. Okay, so the field is growing very rapidly. Okay, let me start my research work on the self-assembled liquid crystalline nanoparticle. You might be wondering what is this self-assembled. So there are certain amphiphilic liquids. When these amphiphilic liquids are, when it is contact with the aqueous domain at a particular temperature, this forms like a unique phase-like structure. We say it's like a liquid crystalline structure. Either it is a liquid or a solid. It is a kind of intermediate. And you can relate this structure, particularly the lipid bilayer, with the liposomal structure, in which there are like enormous lipid bilayer, like infinite lipid bilayers are there, along with the water channels, which has a good potential in encapsulating the drug, preserving the drug, controlling the drug release, and increase the stability. Okay? All right. So this slide depicts the various phases that can be formed by the amphiphilic liquid. Okay? So some of the phases are like the cubic phase. We say cubic, there are different phases like PN3M, IM3M, okay? These are cubic phases, which forms like a cuboidal-like structure. And the other one, which is the hexagonal. These are the two phases widely used in the drug delivery, all right? And the first liquid crystalline phase or the liquid crystalline nanoparticle is originally developed by the amphiphilic molecule, which is monovalent. So monovalent is nothing but glyceryl monovolate, okay? So this particular amphiphilic liquid that forms the critical packing parameter more than one. When we say greater than one, it forms like an inverse micelles, right? So unlike the emulsion, when you add an oil into the water with the surfactants, it forms like a normal one. The CPP is less than 1. So when the CPP exceeding more than 1, so all these structures which increase in the negative curvature result in the formation of various phases. Okay? So we are more interested in capitalizing these various phases for the drug delivery. Okay? All right. And out of the 4 different phases, mostly we use bicontinuous cubic phase, and the second one is the hexagonal phase. All right? So this bulk phase are further broke down into smaller particles, like how you prepare emulsion in the lab. So you add oil, surfactant. The purpose is to solubilize some of the poorly soluble compound into the oil phase, and then you do the homogenizer. It's a simple process. So most of the lipid-based system follow this method. But the important thing is the optimization, and second thing, we need to have a clear understanding on the property of the drug and the type of lipid you are going to select, and what is the target delivery expected. Okay, all right. So these are some of the key features which I have already highlighted: improvement in the solubility, stability, control release. These are the three primary property that is expected from this carrier. Okay, all right. So I'm going to briefly share the research experience, the part 1 and part 2. These are two. The first one is basically a master student project, okay, in which we have enhanced the solubility of dasatinib, an anti-leukemic drug, and which is an anti-cancer drug, based on the quality by design approach. Okay, all right. So whether you are starting an undergraduate project or a postgraduate project or even PhD research work, so first you need to clearly define what is the problem statement. All right. So from there you will derive the objective and then the solutions. Right. So for most of the drugs, the limitations are the poor solubility, and that result in an inconsistent pharmacokinetic parameter, basically the effectiveness. Okay, and so in order to overcome this, we choose certain nanoparticulate system, which we can say like smart delivery system. Okay, there are many nanoparticles out there. So how to choose is depends on the property of the drug. That's why the chemistry plays a crucial role in understanding any aspect, I mean, under the umbrella of the pharmaceutical sciences. Okay, all right. Okay, so this particular research is built on the quality by design. Perhaps some of you might have known what is quality by design. So quality by design, if you want to explain in a layman term, you are building the quality into the product rather than testing the quality of the product. This is a simple definition for you to understand. Right. So how to build the quality into the product? Okay, let me give a simple example for you to understand. If you want to prefer a coffee or a tea, there are handful of ingredients you need. Okay, first is we say like in pharmacy, we say active ingredient. All right. So for coffee, you need a coffee bean powder, then milk, sugar, all right, and perhaps like what is the mixing time, all those things. So here one domain is the material what you need, and the second one is the process what you are going to do to the material in achieving the expected taste of the coffee. If you are simply explaining in the pharmaceutical term, again I take example of an emulsion. So emulsion, you know, the oil phase, the surfactants, all right. So these are the two things with regards to the material, and the third one is the process parameter. All right. So you need to optimize all these things, material as well as the process, which is multivariable. So you have to like consider all these variables, and you have to identify which variable is critically affecting the expected quality. If it is an emulsion, the expected quality is it should be a stable, and if you further delineate that, what the particle droplet or the droplet size you want. Okay. All right. So for this case, I will highlight some of the property of the drug. So it's a BCS classification 2, which is having the solubility problem. All right, and it's an anti-leukemic drug. So for this research, we have utilized quality by design approach using the liquid crystalline nanoparticle. After the optimization, we perform the in vitro evaluation. Okay. All right. So again, so when you clearly define the problem statement, your objective will focus on that. Okay. So the problem statement is the solubility, and the solution is to use this nanoparticle to enhance the solubility based on the quality by design approach, and you have to further validate its effectiveness. Okay. All right. So in the interest of time, the research methodology, I will briefly go through that. It's very similar to how you perform the emulsion preparation. In case of the liquid crystalline nanoparticle, you melt the lipid, you add the drug to the molten lipid, then at the same temperature, you mix the water along with the surfactant, and you do the sonication, you get the nanoparticle. Okay, all right. So for the quality by design approach, you have to start with what is the target quality you needed, all right? So first you need to identify what is the CQA, CMAs, and CPPs. Okay, what is CQA? It's a critical quality attributes, which is derived from the quality target product profile. CMA is the material attribute. Okay, CPP is the process parameter. Again, taking example as an emulsion. The material is the oil, surfactant, water. That's the material attribute. And the process parameter is the homogenizer. So how long you are going to homogenize, at what physical force you need to apply that. Okay, so that's the process parameter. So material attribute and the process parameter together contribute to the CQAs. Okay, and usually it starts with the risk assessment, and finally we choose an appropriate model to optimize that. Okay, all right. So just now I was explaining about the preparation, okay, and for any nanoparticle development, you do all the characterization like particle size measurement, morphological measurement, and finally the in vitro evaluation. Okay, also the stability of the prepared dosage form. All right. Okay, lastly you do the cell culture work. So we have done in the prostate cancer cell line as well as the Kakotu cell line. Okay, let me highlight some of the results what we get. So any formulation development, you focus on the solubility, and you have to choose the excipients. We say excipients, right? First you have to do the solubility analysis. How much amount that can be solubilized in the given amount of the excipients. From there, you have to start the further development. That's the proper way of the development. So, monovolene is all these ingredient is chosen based on the formation of liquid crystalline structure. There are many excipients out there, but it should not affect the formation of liquid crystalline structure. Okay. So, out of the like 6 excipients we have selected, monovolene and 280, monovolene plus 280 has shown higher solubility. So from there we further derived the target product profile, okay, and these are the expected target we fixed, which is the particle size less than 200 nanometer, okay, and with a zeta potential value of -20, and preferably the encapsulation efficiency more than 75%, okay, and sustained release behavior, okay. So the QBD approach, the first, when you have all the variables, the first thing you have to do the risk assessment. Which variable is having higher risk in affecting the quality target, okay. So we have done the risk assessment, and for the quality parameter, which is the particle size, you can see all the variables like lipid concentration, surfactant concentration, sonication time, and the power. It's having a higher impact or the higher risk on that. Okay. So for the other thing is for PDI, only the surfactant concentration will have higher impact. Entrapment efficiency, the drug concentration, and the lipid concentration is the crucial factor. Okay. All right. So we have done the preliminary assessment for all the parameters. So the initial parameter, the initial screening, the QBD done in the two way. First you screen all the variables, so which is going to affect significantly. Okay. So out of which the most significant are the 3 parameters, 3 variables, the lipid concentration, and the second one is the surfactant concentration, and third one is the sonication time. So these are the 3 variables we have taken for the further quality by design method development. Okay. All right. I also got the gift. Okay. So when we run the box Benton design, so we have identified that the two quality parameters is not highly affected except the particle size. Okay. So particle size was the one highly affected by this. All right. So for any model, you have to see whether it is significant or not, and then the lack of fit should be, the p-value should be more than that, which is right. Okay. All right. So in the interest of time, I'm just highlighting the significant outcome of our research. All right. So finally, the three variables are optimized, and out of 72 solutions, we came up with one particular solution, and when we run the experiment with the predicted and the experiment, the deviation is very minimum. So the model which we have chosen is acceptable, and the variability is minimum. Okay. And we have done the further characterization for that. So we have done the thermal analysis. Because you are melting the lipid, it is good to perform the DSE analysis. All right. So which shows the absence of drug peak in that, and you do the morphological characterization. So all the characterization techniques and other result, we got the positive result. Okay. And one of the important characterization technique for the liquid crystalline is the small angle X-ray scattering. So for which actually clearly tell which type of the phases the particle is having. Okay. So we identify this is the cubic structure, and the stability is good. Okay. And then we perform in vitro drug release, which shows sustained release. Right. Okay. And the drug release pattern follows the diffusion mechanism. Okay. So we have come up with all the, we have optimized with all the expected quality targets for this. And finally, we have tested the cytotoxic assay, and it showed a significant difference when compared with the pure compound. Okay. With no significant toxicity to the cell. All right. And cellular uptake assay was significantly higher compared with the pure compound. Okay. So basically, we have achieved all the expected quality. All right, in the part one. So part two, I will not take much time. This one is we have published this paper in Molecular Pharmaceutics, which involves the combination drug delivery of thymoquinone and gemcitabine for the breast cancer, utilizing the same liquid crystalline nanoparticle, but this time is exosome. The previous work which I was presenting is for the cubosome, whereas this one is the exosome. Okay. So we followed the same method of preparation, and we have done the characterization. Also the drug release, sorry, the slide was too much crowded. Okay. And when we do the biocompatibility assay in the zebrafish model, okay, so the result advocates that the carrier which we have developed is having good biocompatibility. This is one of the good model to test the nanoparticle biocompatibility, okay, apart from the cell line. All right. Okay, the cellular uptake was significantly better when compared with the pure compound. All right. So all these studies, part one and part two, we have done up to the in vitro. So further, we are exploring on the active targeting. These are passive targeting, in which only we encapsulate the drug inside the nanocarrier. So currently, we are tagging a ligand on the surface of this nanoparticle, and that research is in the progress by the PhD student. Okay, so we are able to publish when we get some promising result on that. Okay, All right, so these are the future recommendations which follows the in vivo studies that leads to the clinical trial. All right, okay. Going very fast. Okay, so I have to acknowledge all the postgraduate students of my lab. I have collaborators from the University Malaya and University Putra, Malaysia. So they are the collaborators of me, who are also working on the same liquid crystalline nanoparticle. So they will do all the chemistry part, the characterization, and then so the in vitro evaluation is done by me, including the formulation development. Okay. All right. So that's all from my side. Before I conclude, just two positive notes. Okay. So research is all about like how we face the failure, right? So we do multiple experiment, all right? So I think the whole day, all those chemists have shared their experience. They will try with n number of molecules, out of which we get only few molecules click into that. Okay, so we need to keep continuing, all right? So that's the learning we have, all right? So this is the message that is shared by our great president, Professor Dr. Abdul Kalam, all right? And then, all right, so another good note is you must be the change you want to see in the world, okay? All right. So thank you everyone. Sorry, I was a bit rushed because of the time. So I have to go through the things very fast, okay? I'm happy to take any questions from the students or from the speakers.
Yeah, we have time to take couple of questions.
Hello, sir. That was a very nice presentation. I want to ask you actually about what was that biocompatibility assay by zebrafish model?
What was the biocompatibility assay?
By zebrafish.
Okay. So we treat the nano formulations with a zebrafish, which is cultured in the petri dish model, and then it's like the we say hover post fertilization. So we will be examining the morphology of the zebrafish after certain time point, and if there is any deformalities in that, so it will tell you the nano formulation is having certain toxicity. Due to that, there is a deformality occurs in the zebrafish. Yes. It is quite similar to the cell culture work what we are doing.
Hello, sir. I'm Dr. Priya, and I want to ask you, how do you compare this liquid crystalline cubosomes, hexosomes that you are talking about as compared to the conventional dosage form, in terms of if you're talking about efficiency of this liquid crystalline nanoparticles and the cost of these liquid crystalline nanoparticles, and also the production, ease of production also?
Okay, that's a good question. We always, because the industry is very well versed with the production of the conventional dosage form, and
yes,
still the larger proportion comes from the conventional dosage forms,
yes,
right? In terms of the nanoparticle, is one of the slide which was indicated as a milestone. So the field is evolving, particularly in terms of the second question what you have asked on the manufacturing part of that. Okay, so I can take example of the vaccine. All right. So in the short duration, the industry is able to produce the mRNA-based vaccines which utilize lipid polymer hybrid technology.
yes,
All right. Okay. So it depends on the target what we need. If you're preparing a paracetamol, better we go for the tablet formulation. It's simpler to understand.
Exactly.
Whereas the data shows that particularly for the oncologists, where you need to specifically deliver the drug where it is needed and when it is needed. So those challenges, still the conventional dosage form fails to address that. So then that given the opportunity for the drug delivery scientists to focus on all the advanced carrier system. So we cannot generalize, say that, okay, nano is super duper or conventional is bad. We cannot say that. But it depends on the targets what we have and the type of molecules what we choose.
Okay. Thank you, sir. Thank you so much. It was very informative presentation. Thank you.
Sir, I want to ask one more question. Like, how can we say like these cubosomes are means these can be more preferred over NLC or SLN, solid lipid nanoparticles or nanostructured lipid carriers?
Understand. So you want to compare with SLN, NLC?
Yeah.
The
Means how we can decide like for which drug, which formulation should be preferred, because all are lipid formulations.
Okay, right. So again, it's a general way of telling the things. When we do the initial screening on the drug molecule, we never know SLN is better than LCN or liquid crystalline or LC. Okay, but with a clear understanding on the chemistry of the drug molecule. Okay, say for example dasatinib. So the data is not shown. So when we try with all those lipids, solid lipids, the solubility was not that much prominent.
Okay.
Then we work on this carrier. So the target is to enhance the solubility. Each carrier will have its own unique features. Whereas this carrier, when you see the structure, it has an infinite lipid bilayer and then the water samples. So the surface area is really large. And these particular carriers, there are upcoming research on that. Okay. In terms of the preparation method, when you compare with the SLN, NLC, as well as the liquid crystalline, it's more over the same.
Yeah.
It's more over the same. Unlike the liposomes where you, like in lab scale, we do the thin film hydration method. You don't need to go that method. It's like very similar to the emulsion preparation method. Okay. So in a nutshell, I can say that it depends on the drug what we choose.
Okay, means basically we can decide based upon the solubility of a drug carrier in the lipids. Thank you, sir.
Any other question?
Sir, is there any possibility that the nanoparticles may form aggregates or get accumulated in the body?
Okay. Again, it's a general question, right? Whether the nanoparticle can form aggregation or not, right? Okay. So it is good to take example which is already there in the market. Again, today I'm taking all the Pfizer vaccine, one lipid polymer hybrid technology. So when it is injected, the role of this carrier is not to aggregate, all right? It has to shield the mRNA, it has to encapsulate, it has to increase the stability, and then it has to deliver, right? So in terms of the aggregation, so before, that's why the stability is one of the important things. If we see the stability of the nanoparticle in vitro, right? So it is expected that it is translated in the in vivo. It is not guaranteed. So which involves the further studies and then exploration on that. Okay. So again, it's a general question. We cannot say that the nanoparticle will not form aggregate or will form aggregate. Okay. Right. It depends on how we design and optimize the thing.
Thank you, sir.
Hello, sir. Can there be some serious stability issues with this nanoparticles?
Okay, if we experience serious stability issue, then we have to go back to where we start, how we design the thing. Okay, we have to see which kind of stability, whether it is a physical stability is the issue or a chemical stability is the issue. Then we have to figure it out how to redesign the formulation. Okay.
Well, if there are no questions, let us thank Dr. Tyagarajan for his nice presentation. I just want to mention one thing. I always used to wonder why pharmacy has 220 credits, whereas other branches, including technology branches, they have 160 credits. Now I think students, you understood that what we are, where we are, and how much knowledge is required for a drug discovery. In before the lunch session, I think Dr. Vinod Kumar told that there should be a multi-pathway approach. Suppose your scooter is not starting or your car is not starting, you go to the mechanic, he will tell, okay, this some wire was not connected or something is there, the only one problem. But our human body is very complicated. God has made our body in such a way, if one pathway is not working, there are 10 other pathways. So that is how the morning we have learned that there are multiple pathways to travel. And then second part, in the previous lecture, Dr. Raj has explained how complex is the discovery of a molecule. Suppose he has found out some problems also. Suppose he overcomes the problems and he finds out a molecule which is suitable in vitro and animal models. Then comes how to deliver that molecule to the body, and that part has been taken care of by Dr. Tyagarajan. He has that also. There are so many ways to deliver a molecule to the human body, and he has explained only one way, that is the formation of nanoparticle. If your molecule is not soluble in water or it has some other problems, then how to deliver it. So he has given a very nice lecture. I think he has explained some of the techniques of pharmaceutics, QVD and other things. And our students, you must take note of all these things, and let us thank Dr. Tyagarajan for his nice presentation and nice enlightenment. Thank you all.
Thank you, sir.
And then I would like to tell the students that I think today's scientific session is over by now, and then we will have a tea break. But don't forget, there are some students who have made a lot of effort to put posters. Don't forget, go there and see some of your known friends, they might have put up the posters. Thank you very much. Thank you, sir.
Thank you. Thank you, everyone. Thank you.
We thank you, Professor Jagataran Das, sir, for taking the lead as proceedings as the chairperson for this session.
On behalf of everyone here, I would like to extend my sincere gratitude to our esteemed doctors and professors for the insightful presentations. Your dedication to research and education is truly inspiring, and we deeply appreciate the time and effort you have taken to share your expertise with us today.
Thank you for enriching this conference with your knowledge. With this, we conclude this session, and today we look forward to welcoming you tomorrow for session 3 and 4 of day 2 in Canara Hall. We invite you all to join us for high tea at Shivaji Foyer. With this, we officially conclude this event. Thank you for your participation and presence. Thank you.